Ok. Me parece el fin. Cuando me digas, Millán. Ok. Buenos días a todos. Bienvenidos a la Escuela Española de Historia y Arqueología del CSIC en Roma. Me excuso por el director que me da la palabra para que os haga la presentación y para que os haga la bienvenida. Como sabéis, este seminario se va a hacer en distintas lenguas, cada uno hablará en la lengua que más le guste y que más se sienta a gusto, es decir, que no es necesario hacer siempre en inglés, podemos hacer en italiano, habrá conferencias en italiano, en francés, en español, en inglés. Y lo primero que vamos a hacer es un poco presentaros de dónde nace este proyecto, que es un proyecto de 2 años, con ese nombre, NeoNet, Origen, Desarrollo y Consolidación del Neolítico en el ámbito mediterráneo, y que está financiado, voy a poner este momento, porque si no es que no me veo ya a mí, y que está financiado por el Ministerio de Ciencia, Innovación y Universidades del Ministerio Español, del Gobierno español. Lo primero que evidentemente tengo que hacer es agradecer a las instituciones que están detrás de este proyecto. Evidentemente un ministerio, otro es el CSIC, la tercera la Escuela Española, que gracias a ellos hoy podemos estar aquí sentados y podemos discutir tanto los que estamos en presencial como los que están en online. El proyecto tiene, podríamos decir, como 2 codirectores, que son Miriam Cubas y por lo tanto está representada la Universidad de Alcalá, y Millán Mozota a través de la institución Milà i Fontanals, de la cual vengo yo también, pero por lo tanto está representado también Millán. Y por último, en este caso, en esta segunda reunión, porque esta es la segunda reunión que hacemos, hemos puesto como colaborador también al Museo de la Civiltà, Luigi Pigorini, porque la verdad es que nos han ayudado a hacer, hay una parte que es la visita a las instalaciones del museo y a ver los materiales de la marmota, que ya vimos ayer, y por lo tanto hemos considerado que era interesante que también estuvieran como parte de las instituciones. Bueno, primero agradecer a Antonio por habernos dejado este espacio y haber apostado siempre por nosotros para poder hacer esta línea de prehistoria dentro de la escuela. Yo creo, no estoy muy seguro, Antonio, igual digo una barbaridad, pero ha habido prehistoriadores, pero creo que es la primera vez que hay una línea de prehistoria dentro de la escuela. Por lo tanto, el hecho de apostar por nosotros para esta nueva línea, yo se lo agradezco personalmente. También a todos los colaboradores que están tanto en la escuela, es decir, desde Antonio como el máximo representante hasta la señora de la limpieza. Todos los son tanto investigadores, cuerpo administrativo, contables, bibliotecas, han hecho posible que hoy pudiéramos hacer esta reunión. A todos los colaboradores de la red, que ahora os haremos una pequeña presentación de quién son esos colaboradores de la red. Y a todos los que estáis presencialmente o conectados online, que vais a ver las conferencias online. Y que una cosa muy importante a través de las conferencias online es que todos los que están en YouTube pueden hacer sus cuestiones a través de YouTube. Es decir, no solo son cuestiones a nivel presencial, sino todas las personas que estén conectadas a través de YouTube pueden hacer sus cuestiones y después las podemos contestar. Para hacer 4 datos sobre el proyecto, está en la convocatoria de acciones de dinamización red de investigación, con ese número, y como os he dicho, ha tenido 2, este es el segundo seminario, de hecho solo va a haber 2, pero el primer seminario fue el año pasado y se hizo online, porque evidentemente por la pandemia estuvimos obligados a hacerlo online, y afortunadamente, hubiéramos querido que fuera presencial con la mayor parte del grupo, pero evidentemente es imposible. Es decir, que hay compañeros y compañeras de distintos países que no podían venir por su situación sanitaria o por su reglamentación sanitaria en sus países. Pero en todo caso, están online también. Bueno, esta es la organización. Yo realmente no sé si debería ponerme ahí, el coordinador de la red soy yo, porque es el que firmé al final el proyecto, pero la ideóloga del proyecto es Miriam Cubas, aunque se esconde por allí, nunca quiere aparecer, es Miriam Cubas. Ella fue la primera que me llamó para decirme hay que pedir este proyecto, pero como en ese momento no lo podías pedir, pues al final salgo yo en los papeles, pero la ideóloga es Miriam Cubas. Y con ello quiero decir que le paso el testigo ahora, que ella sí que lo puede pedir, le voy a tirar el testigo para que en la próxima red pues lo pida ella, así sale su nombre. Porque entonces uno tiene la sensación que se ha aprovechado un poco de la jugada. La estructura de la red está, para los que no la conocen, tiene dos partes. Una que son los investigadores e investigadoras que hemos invitado porque trabajan en determinadas zonas, especialmente era de la península ibérica, pero después hemos invitado a investigadores que vienen de otras zonas del Mediterráneo. Aquí los tenéis. No voy a citarlos todos, pero bueno, como queda grabado, sí, podéis verlos uno a uno. Y después hay, dentro de la red, a investigadores e investigadoras que lo que hemos hecho es invitarlos por la temática en la que trabajan. Pues había gente que estudia el clima, la fauna, las semillas, la tecnología. Es decir, que no es solo por la zona geográfica, sino también por la temática. Estos dos grupos fueron los que básicamente formaron parte de la primera reunión, que fue una reunión, como he dicho, online, y que básicamente se invitó a la gente de la península ibérica a hacer la primera reunión. Que, como también he repetido, nos hubiera encantado reunirlos a todos, porque presencialmente es mucho mejor, creemos, pero que las condiciones de la pandemia nos impidió absolutamente reunirnos presencialmente. Y en la segunda reunión que hoy tenemos aquí, la idea era que aquellos investigadores extranjeros que formaban parte de la red participaran en esa segunda reunión. Y ha dado la casualidad que en esta segunda reunión, pues volvemos por las condiciones de la pandemia, ha sido posible que parte de esos investigadores hoy puedan estar aquí presencialmente. Y algunos están online. Y también, aprovechando que estábamos aquí en Italia, pues hemos invitado, aunque no formaban parte inicialmente de la red, hemos invitado a algunos investigadores italianos, que para ellos era bastante sencillo, era bastante fácil venir aquí a Roma, a que presentaran algunas de sus trabajos. Como os he dicho, la estructura era esa: áreas geográficas de la península ibérica, áreas del Mediterráneo. Por eso hoy, bueno, durante estos 2 días va a haber investigadores que nos hablen de Grecia, de Macedonia, de Croacia, de Italia, ¿de acuerdo? Y después las cuestiones más de metodología o de temáticas más concretas. Ahí tenéis las zonas. A cada zona se le asignó un investigador, como uno o varios investigadores principales de esa zona, y podéis verlos aquí. Estas eran las del primer seminario, por lo tanto eran sobre todo de la península ibérica. Y después los grupos de trabajo, como hemos dicho, que eran pues los que se dedicaban al grupo de los domésticos, clima, instrumentos, cerámica y antropología. Esos fueron los grupos que realizamos. En este segundo seminario, pues hoy lo vamos a ver, pero como aquí os enseño un poco, como hemos dicho, hemos invitado a aquellas personas que no eran, o investigadores que no eran de la península ibérica y por lo tanto que iban a participar en este segundo seminario. Y en Italia tenemos Mario Mineo y nosotros que trabajamos con él, Carmine Collina, Goce Naumov con Macedonia, Kostas Kostsakis con Grecia, Bogdana Milic con los Balcanes, Antonio Carballo con Portugal, Staso Forenbacher con Croacia, y como una invitación especial hemos hecho a Primitiva Bueno también, que nos va a hablar del sur de Europa. Y en aspectos metodológicos, pues a Oliver Craig con los residuos de cerámica, Eva Fernández con el ADN, Amy Bogart con los análisis ecológicos, Tomás con el tema de dataciones, que es un trabajo que estamos haciendo desde el primer seminario, y Manuel Rojo y sus compañeros con el tema ambiental. Rápidamente se creó una red. Lo que queríamos era una vinculación cercana con los investigadores y con las instituciones que tenían, que les representaban. La red tiene por objetivo el intercambio de conocimientos científicos y también la oportunidad de que tanto los investigadores de la red como estudiantes pudieran ponerse en contacto con la gente de la red para acceder o de alguna manera, si estaban interesados en participar o en sus tesis o en ir a sus laboratorios, etc. Esta era la idea de la red. En este segundo seminario lo que hemos hecho es, ahora yo estoy haciendo la presentación de la red, después vendrán las conferencias invitadas, en varios momentos nos vamos a reunir la gente que está en la red aquí para tratar la red y ver qué futuro tiene la red, si seguimos o no seguimos. Yo espero que Miriam quiera seguir. Después al Museo de la Civiltà ya fuimos ayer y al que desde aquí quiero un poco agradecer a Mario Mineo, que nos abrió las puertas, pero también a su directora, que no quiero decir mal el nombre, que es Loretta Paderni, que nos abrió las puertas del museo, a Massimo Sana, que es el director general, que por lo tanto tiene que ser consciente que estuvimos ayer allí y que nos abrieron las puertas. Y de alguna manera quiero hacer un recordatorio al antiguo director del museo, que falleció de COVID en noviembre, que es Filippo Maria Gambari, que conocía la red y que evidentemente siempre apostó por que nosotros pudiéramos hacer este seminario y que pudiéramos ir al museo. Bien, como os he dicho, la sesión es en línea abierta y presencial y la idea es poner al final en puesta en común las futuras iniciativas. Entre las acciones que hemos hecho en estos 2 años, evidentemente al estar en pandemia hay cosas que prácticamente no hemos podido hacer, que eran objetivos y que no hemos podido hacer. Por ejemplo, había una idea que era hacer cursos de formación específica a nivel universitario en algún sitio, algún tipo de curso. Miriam había pensado uno de cerámica, por ejemplo, pero se nos ha sido imposible por el tema de la pandemia. Pero sí que de una manera u otra hemos contactado con los investigadores, se han impulsado nuevos proyectos, algunos estudiantes han contactado con nosotros y han podido acceder o los hemos puesto en contacto con investigadores para hacer sus investigaciones. Es decir, que yo creo que los puntos 2, 3, 4 y 5 sí que de alguna manera los hemos llevado a cabo. Después había otra de las acciones a nivel de museos, que era hacer conferencias y charlas también en los museos, pero ha sido imposible. Es decir, como no podemos trasladarnos, pues ha sido imposible. Y lo que hemos hecho es elaborar unas guías con el estudio de determinados materiales y temáticas y que publicaremos y que de alguna manera será el elemento que llevaremos que mandaremos a los museos para solventar esa parte que nos hubiera gustado, que es llevar alguna conferencia, alguna charla a distintos museos para que supiéramos qué estábamos haciendo. Y con el tema de la publicación en general, de la divulgación para públicos, pues sí, se diseñó una página web. Se han hecho, gracias a Millán Mozota y un proyecto de la FECIT, alguna representación teatral y hemos participado en eventos nacionales e internacionales a nivel divulgativo, pero, por ejemplo, una cosa que queríamos hacer, que era una exposición que pudiera ser trasladada a distintos centros, eso no se pudo poder hacer. Y lo que hicimos al final fue que en una exposición la idea era hacer una serie de pósters de cada una de las temáticas y esos pósters, como no se pudieron llegar a hacer a nivel presencial, lo que hicimos fue ponerlas en la web para que todo el mundo pudiera tener, por así decirlo, que hubiera una exposición virtual. Y nada más, doy las gracias intentaremos que no haya ningún problema a nivel online. Y si no me equivoco, el programa, el primero que habla es Carmine Collina, al que agradezco mucho que haya venido. Vamos. Un segundo, Carmine.
Allora, Juan, innanzitutto grazie per l'invito et je vais prendre à la lettre ce qu'a dit Juan et donc je vais parler français, mais peut-être vous allez trouver des diapos en anglais ou en français et je m'excuse, mais je vais parler en français. Alors, ce que je vais présenter aujourd'hui, c'est un travail qui abordait la question de l'étude technologique du Néolithique ancien et du second Mésolithique, du Mésolithique final en Italie du Sud. En particulier, on a travaillé sur l'étude des séries lithiques qui permettent d'encadrer le transfert, les dynamiques de transfert culturel entre 7e et 6e millénaire avant Jésus-Christ calibré. Les objectifs généraux de ce travail, c'est donc la description des chaînes opératoires et des techniques, et puis l'identification des traditions techniques mises en place, et puis on a essayé de reconstruire des processus évolutifs entre le 7e et le 6e millénaire en Italie du Sud. Bien évidemment, on a choisi des séries lithiques qui gardent, comme on dit, une haute fiabilité stratigraphique. Donc, on a fait un tri où il s'agit des collections qui ont été fouillées dans les années 80-90 du siècle dernier. Et donc, on a choisi, on a fait un tri sur la fiabilité stratigraphique des sites. Le point de départ est donné sûrement par la grotte de Luzzo, sur lequel on a étudié la série des tranchées F et M, des tranchées de fouilles F et M qui gardent la séquence du Mésolithique final, et puis toute la séquence néolithique de la grotte de Luzzo. On a travaillé sur l'Afronico 3 pour le Mésolithique, et sur les différents horizons culturels néolithiques, on a choisi des sites pratiquement dans toutes les régions de l'Italie du Sud. Les questions qui ont donné la démarche à ce travail, c'est donc, est-il possible de donner une valeur culturelle à la variabilité des faits techniques? Et puis, la reconstruction des rapports entre les derniers chasseurs-cueilleurs et les premiers paysans, et bien sûr, la grotte de Luzzo nous donne la possibilité de travailler sur ce sujet. Et puis, la possibilité de reconnaître une variabilité techno-économique dans le système de débitage du second mésolithique et puis du néolithique ancienne en Italie du Sud. On va faire rapidement un résumé des horizons culturels qui, traditionnellement, dans l'histoire de la recherche, ont été impliqués sur le sujet de la néolithisation dans le sud d'Italie. Donc, pour le mésolithique et l'épipaléolithique, il y a le romanélien, que c'est une fasces épipaléolithique localisée surtout dans les Pouilles, l'épipaléolithique indifferentiato, identifié par Martini à la fin du siècle dernier, et puis la série sovétérienne castelnovienne, caractéristique de la Méditerranée. Et pour les néolithiques, il y a tous les différents horizons de la céramique imprimée jusqu'à céramique peinte et gravée. Ça, c'est la distribution géographique des sites les plus importants pour les fasces mésolithiques. Je vais rapidement présenter le cadre pour les sites identifiés, les sites principaux identifiés en Sicile. Il faut noter que le seul site qui a une séquence fiable qu'on peut attribuer au Castelnoviano ou au complexe à lame et trapèze dont on va parler depuis quelques minutes, c'est la grotte de Luzzo. Dans la littérature, on avait posé un sujet, on avait posé une question, c'est-à-dire la permanence des traditions et des traditions lithiques épipaléolithiques dans les séries néolithiques, et ça, on l'avait soutenu surtout avec des données typologiques jusqu'au début des années 10 de 2000. Mais en effet, soit l'étude chronologique, soit l'étude des séries lithiques ne permettent pas de confirmer des permanences épipaléolithiques dans les séries néolithiques. Et ce qui est bien sûr évident, c'est que dans le 7e millénaire, il y a quelque chose qui représente une rupture culturelle, une rupture technologique. Et donc, c'est à partir de cette rupture qu'il faut se poser la question de la continuité avec les complexes, entre les complexes méso et néo. Ça, c'est un diapo pour montrer les horizons céramiques qu'on va traiter dans nos séries, dans les séries lithiques qu'on a choisies. Et ça, c'est ce sont les sites qu'on a abordés pour la Sicile en distinguant un horizon impressa pre-Sentinello et un horizon néolithique Stentinello. Quand on a fait le tri des données chronologiques les plus fiables, bien évidemment, ce qu'il y a, c'est un hiatus, ce qu'on voit, c'est un hiatus chronologique des dates les plus fiables entre la fin du 7e et le début du 6e millénaire, et puis en début de la céramique imprimée archaïque à environ 5900 calibrés. Et puis, une diffusion de la céramique imprimée évolue ou impresso cardial sensu binaire à partir de 5800, grosso modo. Et ici, on présente l'approche méthodologique qu'on a suivie. Donc, la démarche conceptuelle est donnée par la chaîne opératoire et les outils des travail sont l'économie, les conseils de l'économie de la matière première, l'économie du débitage, ce qui nous permet de, comment dire, encadrer les industries lithiques dans une optique territoriale et bien sûr dans une approche synoptique. On a suivi différents moments dans notre méthode. Puis, on est parti par l'étude de l'analyse, par l'étude des matières premières, donc une approche pétrographique, et puis l'approche technologique par le classement des produits de débitage, la segmentation de la chaîne opératoire, donc le diagnostic des techniques, et l'identification des méthodes de débitage, puis l'analyse typométrique et une analyse typologique de l'outillage qui a essayé d'intégrer les critères technologiques dans la description des morphologies des outils. Il y a 4 ans plus ou moins, on a commencé à faire des études fonctionnelles associées aux bases de données qu'on avait déjà. Donc, et on a commencé à travailler sur la partie tracéo sur les sites de la Starza et sur les sites de la Tronico 3. On a fait ça avec des collègues français, Colaguerre et Bernard Garcin. Donc, on va partir avec une synthèse des systèmes techniques à la grotte de Luzzo et à la grotte de la Tronico 3. Ce qu'on a vu, c'est que la pression et la technique de détachement utilisée pour la production de lamelles prismatiques, ces lamelles sont destinées à être transformées en trapèzes. Et la méthode est caractérisée par une série de cours de détachement de 6-7 lamelles qui sont débitées selon un rythme convergent sur des surfaces à faible convexité transversale. Et les plans de pression sont remodelés par un facettage systématique des plateformes de pression. La variabilité des chaînes opératoires est liée aux possibilités de réorientation de débidage offertes par la matière première. Et bien évidemment, ces modes de débidage ne produisent pas de lamelles régulières qui a en faible proportion. Donc, la pression est peu visible si on retient les caractères les plus emblématiques, je pense au parallélisme ou à la régularité d'énergie. Le détachement des talons facettés sur pédant qui s'étalent sur la largeur des pièces assure la reproductibilité des processus de production. Donc, à la fin, ce sont surtout les produits centraux qui sont prélevés pour la production des trapèzes. La pression a été identifiée, comme on dit, par des facteurs positifs, c'est-à-dire observation de présence, et négatives, observation d'absence, qui permettent d'exclure la superposition avec la percussion en direct. On ne va pas rentrer dans le détail, si vous voulez, on peut le faire après dans la discussion ou dans le débat, mais je vais remarquer que ce modèle qu'on a reconstruit, à la grotte de Luzzo, on le voit aussi dans les phases néolithiques, soit néolithique impresse archaïque, soit dans le néolithique scientifique. Donc, le grotta de Luzzo représente sûrement le point de départ de ce travail. La séquence culturelle est issue de plusieurs tranchées fouillées à l'intérieur et dans le talus à l'extérieur de la grotte. Et dans ce schéma, on voit la tranchée A à l'intérieur qui garde la partie plus ancienne du dépôt, de l'épipaléolithique jusqu'à la phase mésolithique plus ancienne, et puis les tranchées à l'extérieur, la F et la M, qui gardent la série, la séquence entre seconde mésolithique et néolithique. Dans des travaux précédents, on a pratiquement reconsidéré le terme transition qui avait été initialement utilisé pour décrire, pour indiquer les niveaux 14 ans des tranchées F et M. Donc, on a reconsidéré le terme. Il y a plusieurs éléments qui nous permettent d'exclure le concept de la transition et, en effet, en gardant aussi les données chronologiques, c'est qu'on voit que la date de la tranchée F de la grotte de Lutz représente aujourd'hui encore une des dates les plus anciennes pour le complexe lame et trapèze de ce millénaire dans la Méditerranée du sud et de l'ouest. Et ça a été confirmé par des travaux récents, un travaux qu'on va publier dans les prochains mois et un travaux qu'on a publié dans le 2018, 2019. Une nouvelle série de dates sous des fossiles humains et sous des coquillages ont permis de confirmer cet horizon très ancien pour la grotte de Lutz, pour le second mésolithique de la grotte de Lutz. Donc, c'est à partir de ces données qu'on a attribué la phase initialement définie des transitions, tout à fait, pour castelnovian ou complexe lame à trapèze de la Méditerranée. La production des trapèzes du mésolithique final, une phase néolithique, est une production caractérisée par la production des lamelles obtenues par la pression, et les éléments diagnostiques qu'on vient de décrire s'inscrivent dans une méthode d'épidage et dans une chaîne opératoire clairement conditionnée par le type de support, c'est-à-dire des galets de dimensions plutôt modestes, des petits nodules, galets ou petits nodules. La mise en forme est très faible. Il y a une ouverture pratiquement directe du plan de pression. On observe très peu des éléments à crest. Il y a des lamelles corticales. Le plein d'évitage est représenté par des lamelles trapézoïdales surtout. Et ici, les lamelles sont transformées surtout en trapèze. Et puis, il y a une autre segment de la chaîne opératoire qui est dédiée à la réutilisation des éclats de mise en forme. Il y a la technique de microbourrin qui intervient, et on a déjà parlé sur le procédé technique, on a déjà dit. Donc rapidement, c'est ça, ce qu'on a fait sur les matières premières dans le promontoire de San Vito lo Capo. On a identifié pratiquement les formations en silex, les formations en rhyolite, soit en position primaire, soit en position secondaire. Surtout, c'est ici dans le estuaire, sur la rivière du Guidaloca, qu'on a trouvé une grande partie des matières premières utilisées dans les séries lithiques. Ce sont des exemples des types qu'on a identifiés. Et si on voit les histogrammes de distribution des matières premières, on voit une grande régularité entre les trois horizons qu'on a étudiés, donc seconde mésolithique, néolithique ancienne, impressa, et néolithique ancienne, sentinelle. La production des trapèzes, vous la voyez dans la phase mésolithique, est représentée par des lamelles avec une grande régularité des nervures. Et donc, ce sont vraiment les éléments centraux qu'on disait avant. On le voit aussi sur cet exemple de microbourrin distal appliquant triade virtuelle à droite. Et ça, c'est un exemple d'un microbourrin proximal avec une lèvre d'arrachement bien exprimée, un bulbe déturé, le talon facetté large. Donc, c'est un peu emblématique des procédés techniques qu'on a identifiés. Un autre exemple sur cette microbourrin. Ça, c'est le déroulement du débitage sous ces nucléus. Ça, c'est un nucléus de la phase pré-sentinelle. On est passé au néolithique, sur lequel on voit la même méthode qu'on a décrite, et on le voit aussi sur la phase pré-sentinelle, sur ces nucléus qui gardent encore la surface originale du galet. Encore, ça c'est un exemple de lamelles dans la phase néolithique. Et je vais présenter quelqu'un des trapèzes néo, parce qu'ici on voit la variabilité typologique, morphologique, c'est-à-dire les trapèzes mésolithiques sont courts et symétriques. Les trapèzes néolithiques sont plus allongés, plus asymétriques, et gardent souvent une grande facette d'épicantriède. Ça, c'est encore un exemple des niveaux provenant de niveaux sentinelles. Donc, la synthèse qu'on peut faire, c'est qu'on voit dans le second mésolithique une pression à la main ou peut-être à la petite béquille, une localisation des surfaces de débitage sur la face large et étroite des supports, des trapèzes courts et symétriques. Ces modèles, on voit que la stabilité de ces modèles pression trapèze dans le néolithique, avec des éléments de rupture qui sont des ruptures techniques qui se perdent pratiquement rapidement dans la série lithique, et puis une variabilité liée surtout à les trapèzes qui sont plus asymétriques, plus gros, et à la variabilité des modes de production par la pression. Dans le néo, on voit des modules un peu plus forts, un peu plus importants. On va passer à la grotte de la Tronico 3 en Basilicate. On a choisi de travailler sur le niveau 41-44 de la séquence, qui est la partie mieux gardée de la stratigraphie. On est dans le secteur D, mais le projet était et est de travailler sur toute la série mésolithique et puis passer à la série néolithique. Il y a en ce qui concerne les matières premières, ce sont surtout des matières premières locaux, des matières premières locales, avec un type qui a été par nous associé au gargano, mais on n'est pas sûr encore de cette attribution. Ce sont des lamelles provenant de la Tronico. On voit la même, un peu la même morphologie des lamelles de Luzzo, avec plus des éléments à talons torsés, comme on le voit ici, qui sont probablement attribués à cette variabilité de la réorientation des nucléus, encore de lamelles. C'est un détail de diagnostic des techniques qui est très proche à ce qu'on vient de décrire pour la grotte de Luzzo. Je vais vous montrer surtout l'absence d'abrasion de la cornice et le facettage des nucléus. Donc, un lamelle encore de plein d'épitage qu'on a attribué au silex garganique que je vous disais avant. Oui, la variabilité technique qu'on voit à la Tronico, parce qu'il y a aussi des lamelles de plein d'épitage qui ne sont pas associables avec la pression. Le choix des lamelles brutes de dépidage avec des traces macroscopiques d'utilisation montre qu'il est associé surtout aux lamelles plus allongées. Ça, c'est le cadre des trapèzes courts symétriques qu'on disait avant. Ici, on a fait des études fonctionnelles à l'aide avec Bernard Gassen et Collagueré, et on a vu que les trapèzes sont surtout à tranchant transversal. Donc, en passant au 16e millénaire, rapidement, on voit donc la gestion complexe des matières premières, une gestion complexe du dépidage, la segmentation des chaînes opératoires dans le territoire, et puis une sélection des matières premières, c'est-à-dire, on voit que sur les matières premières locaux, il y a surtout un dépidage occasionnel et peu élaboré, alors que les lamelles de plein dépidage sont introduits les lamelles sous le silex exotique potentiellement garganiques sont introduits sous forme de lames lamelles de plein dépidage. Et ce sont les diagrammes sur lesquels on a travaillé, Rendina la stazza et Ripatetta dans les différentes phases, et on voit le même comportement de sélection des silex locaux et silex exotiques et organiques que je viens de dire. Bon, ce sont des exemples de silex de Ripatetta ce sont des types, c'est là, le silex majolica, et puis des silex locaux. La même chose pour la starza. Puis le travail qu'on a fait, mais on ne va pas rentrer dans les détails, pour associer la pétrographie qu'on a trouvée, les types pétrographiques qu'on a trouvés dans les sites, avec les gîtes connus dans le Gargano. Et ce qu'on a vu, c'est qu'il y a surtout le calcaire de type maiolica, schistia fucoidi, et on voit la même distribution à Dipatetta, qui est très proche, relativement très proche du Gargano, mais aussi à la Starza, qui est plus loin, en Campanie, donc plus loin du promontoire. Ce sont des exemples de la variabilité à Dipatetta, sous silex exogènes et silex locaux. Des nucléus par le niveau du second néolithique fase guadone di Dipatetta. Les ensembles de lames du secteur NEO de Rendina, avec les niveaux les plus anciens de la série des séries néolithiques qu'on a étudiées. Et bon, ce qu'on voit sur Dipatetta et Rendina, bien sûr, il y a toute la variabilité des modes d'épidage, des méthodes d'épidage, de plusieurs modules pour l'âme, lamelle, mais ce qui représente la chose la plus intéressante, c'est que à Dipatetta et Rendina, on a reconnu la production d'une pression nullière, donc qui tout à fait aujourd'hui, c'est la plus ancienne production pour l'Italie du Sud, mais bien sûr pour l'Italie. On l'a reconnu à Rendina dans les niveaux impresses archaïques et à Dipatetta dans les niveaux guadone de la ceramica impressa guadone. Dans la Starza, il y a une production des lamelles plus occasionnelles sur le silex local, des produits de provenance locale, et une production plus spécialisée sur le silex exogène. Et cette production est associée à des traces fonctionnelles bien évident, et représente une modalité de la pression plus développée par rapport aux industries de Luzzo et de la Tronico 3. Dans les niveaux les plus anciens, on voit cette production de lames avec lustre et microdenticulés. Ce sont des modules stylistiquement très réguliers qui montrent cette trace d'utilisation. Donc, ce qu'on voit en synthèse, pour le mésolithique final, il y a une pression d'épitage frontal à faible convexité transversale, une localisation des surfaces d'épidage sur la face large et étroite des supports, des trapèzes courts symétriques ou légèrement asymétriques. Cette tradition lithique s'inscrit dans la problématique de la diffusion du complexe à lame et trapèze dans la Méditerranée. Donc, il y a plusieurs hypothèses qu'on avait considérées. Personnellement, au début, j'avais pensé à la liaison entre la grotte de Luzzo et le cap Sienne supérieur de l'Afrique du Nord. Mais les travaux récents sur la chronologie sembleraient confirmer la diffusion par l'Est dans le 7e millénaire de ces complexes. De toute façon, le niveau, le marqueur chronologique, la date de Luzzo, c'est encore très ancienne par rapport aux dates provenant de l'Adriatique ou du nord de l'Italie. Donc, à mon avis, il est encore ouvert la question. Peut-être il faudrait faire de nouvelles dates sur les Cap Sienne, sur l'Afrique du Nord, pour voir si effectivement la grotte de Luzzo est plus ancienne ou est contemporaine ou est plus récente. On voit la permanence de cet aspect technologique dans les séries du 16e millénaire. Et puis, on l'a vu dans la série d'Escamous, on l'a vu dans des aspects de la série de la Starza. Et puis, dans les néo, il y a un développement technologique. Nous, on a enregistré surtout l'investissement technique sous la pression. Donc, on a vu comme cette concept technique a développé jusqu'à l'introduction de la pression houlevière. Donc, il y a des éléments de continuité qu'on voit bien sûr dans la grotte de Luzzo, qu'on voit dans quelques séries de pouilles qu'on a vu en partie dans la Starza, qu'il faut vérifier à la Tronico, et puis il y a des éléments de rupture culturelle qu'on enregistre sur le site de céramique qu'on a considéré. Avec ça, j'ai terminé. Je vous remercie pour l'attention. Ok. Ok.
No, because we have to speak.
Buenos días a todos. Buongiorno a tutti. Ok, grazie per le invitazioni oggi. Mi senti bene?
Because we asked everything.
Va bene. Devo avanzare in questo. Ok, grazie per l'invitazione. Io vado a continuare in inglese. Va bene? Speriamo. Okay, so we were going to talk about the early Neolithic of Southern Europe and some of the work on volcanic residue analysis that we've been conducting with Miriam. And what we thought we'd do today, we'd do a joint presentation, so very ambitious. And I'm going to start and give a brief introduction of what organic residue analysis is. And then I'll say a little bit about some case studies we've been applying to the Mediterranean region. Then I'll pass to Miriam so she can talk about her study that was conducted a few years ago along the Atlantic coast of Europe. And then I'm going to really just throw in some ideas of what we want to do in the future. So very quickly, organic residue analysis is the analysis of pottery to work out their use using an organic approach. We're looking at molecules that get absorbed into the ceramic surface. And it works really because particularly prehistoric ceramics are very porous and they act as molecular sinks. They suck molecules into them, which we then can analyze. And the majority of work that we've been doing involves looking at the lipids. So those are the fats, oils, and waxes that are present in the natural environment that get used in the pots and get absorbed into the vessels. And to do that, we use a method mainly called mass spectrometry or gas chromatography mass spectrometry, where we separate out the extracts and we analyze these individual compounds to try and work out the kind of foods or other products that were present in the pottery. More recently, one of the big advances in this approach has been to actually use an isotopic approach. So we're measuring also not just the molecular constituents of the pot, but also the isotopic composition. And for that, we can get a lot more information on the types of products that we use. I'll explain a little bit about that. A new thing that we're developing is hydrogen isotope analysis, which can give us information about the seasonality of the climate of the products from the organic residues that are in the pot. So this is coming online. It's becoming quite exciting. And you might have seen more recently as well, our colleagues in Bristol have developed a really important approach for dating, actually dating the molecules inside the pots to get direct dates, AMS dates, carbon-14 dates from the use of the vessel. And you can imagine why that's going to be really exciting in the future, given the difficulties of directly dating pottery. What can organic residue analysis tell us? Well, the majority of studies have been really focused upon paleo diet, what was eaten in the past. And this is particularly important for identifying products such as dairy foods, which we'll talk about in a bit, because they're very difficult to characterize using other approaches. Work is not all about food. There has been an identification of other types of products to look at natural products and how they were prepared and what they were used for. But I think what's really been missing, and one of the pleasures of working with Miriam, is to try to integrate the kind of organic signals with the artifact studies as well. So the manufacture, the discard of the artifacts, and trying to integrate this analysis together to look at holistically at how an artifact was used throughout its use life, to reconstruct an artifact biography. And then what we've also been really interested in is actually thinking about, well, it's not all about diet. What goes in the pot is also culturally mediated. And I think cuisine is a very good word to kind of, as an interpretive tool to explain what we're actually finding, how people are preparing and consuming foods. So what can we identify through lipid residue analysis? Well, a lot of focus is on animal products, and we'll talk about that. And how basically we can identify ruminant products. So those, products meat from cattle, from sheep and goats. Non-ruminants, which would be mainly from pigs and other domesticates. Dairy products, as I said, has been identified. Aquatic animals, we have a very good way of identifying aquatic foods. Plants has been a real challenge, and that's what we'll talk about in a bit. It's really difficult, and I think this is where the field is really moving to try and identify plants. And then alcohol, and I'll try and touch on that at the end. It's a big area. We always get asked this, can we identify beer or wine in pottery? And I'll try and say something. So how we do this analysis, basically very simply, these foods get cooked in a pot, they absorb in, or they get encrusted on the surface. And then in the lab, we do the methods to extract them and do the analysis. A little bit about the sampling. We usually take a small scraping from drilling from the inside of the vessel. So I've got to show this so you can see how destructive it is. We don't destroy the outside, it's usually just from the inside. Some of you would have seen some of the samples that we've taken yesterday from La Marmotta. We can also work with the food crusts, these charred deposits on the inside of the pots, which are potentially useful. And more recently, as well as doing the organic analysis, we've been working with colleagues who have been looking at the plant microfossils and other kind of food remnants macroscopically visible within the food crust. So that's another complementary method we can use. And then more recently, my colleague Jesse Hendy has been working on analysis of the calcite deposits that we find inside pots. You might have seen these on some of the vessels, these kind of calcio, these carbonate deposits on the inside. And they're also, if they're from the use of the pot, preserving proteins really well. So they're a really important source for the future. So very quickly, how does it work? We have two approaches. One is using chemical biomarkers. That's if we find a particular chemical, we can be sure that that product was prepared in the pot. And for that, we have several. We can identify millet, for example, milio, but we've also done a lot of work to identify fish. So we can identify fish products, fish processing very easily, aquatic products, I should say, to be more precise. But more generally, we're using a kind of continuous variable approach where we're making measurements of the isotopes of the different compounds and trying to distinguish different products. And very quickly, I'll say about that. This is how it works. So we are measuring the isotope values of the main facts within the pot. And you can see this is the chromatogram. And these are two facts that we find very commonly. So even when we have very little else preserved, we can usually work with this material. We measure the values, and then we can plot these, and we can distinguish different products based upon their isotopic composition, for example, dairy or marine, sometimes plant or ruminant animals. So it works quite well. So to give you an example, and I think it's a good time to start looking at Mediterranean pottery. Not that much has been done, I would say, compared to other parts of the world. And if we look at the kind of impressed cardial complex, which we've been talking about today, very well understood in terms of dating, and we heard about the new dates that have come out from work from people like Didier Binder. So it's an exciting time to be working on this material. But as I said, very little has been done. I'll give you a quick flavor. So this is a study that we did some time ago now, before the new dating, in fact, and you know, we didn't have that good a chronological control, but we managed to get a whole load of sites across the northern Mediterranean basin together and analyze a whole load of vessels to work out their use, including some of the sites we were talking about earlier today. And what we found in this study, this large study, was actually a lot of variability in the use of pottery. Now, what I've done here is plotted those isotope values, but looking at the difference between them. And that can really help us distinguish dairy foods from ruminant animals from non-ruminant. So in this bottom area here, this is where the ruminants, the dairy products, the milk or the dairy products should fall. And you can see in the Levant and Anatolia, very few dairy products. The majority is ruminant meat, probably from domesticates, which were there at the time. And then we get a big change when you start to amalgamate the sites from North Anatolia. We get a lot more dairy production. But when we move to Greece, very little at all. So we're not seeing a standard process of how pots being used. We're seeing this regional variability. And then when we move to the central and the western Mediterranean, perhaps what we're most interested today, we can see that actually, also central Mediterranean, we do have a lot of dairy products. Western Mediterranean, very little studies that have been done. Dairy is there, but it's also quite variably, and it's variable site to site. But you can see there's been a few more studies since then. There's been work done in the south of France, the site of Pendimoun, from Léa Drieux, who's done some work on the residues, also found very little dairy at this site. And there's been some work done in Croatia as well. But apart from that, the amount of residue analysis done in the Mediterranean is very little. Okay, Miriam.
Yeah. In this context, we started some years ago a project that's focused on the pottery in the Atlantic coast of Europe. As you well know, the Atlantic coast is quite well known in the archaeological world, especially for the Mesolithic archaeological sites. So we have a very strong Mesolithic record in the area, mainly in shell middens. And of course, it's a coastal area. So from South Portugal to the north, to Normandy, more or less, it's a quite huge geographical area characterized size by these Mesolithic groups all along the coast with the shell village. However, in this area, the early Neolithic is not really well represented. So in the context of the Neolithization, the Atlantic coast is one of the later areas in the European continent, and the early pottery in this area is not huge. So it was a quite challenge just to choose the archaeological sites and how to decide how to analyze these assemblages. So we decided to carry out a project focused on this area just to see the use of the early pottery all along the coast, especially for this strong Mesolithic tradition before, and especially for characterized for this coastal character. So the earliest pottery in the area, especially in the south, is characterized by the presence of cardial, especially in South Portugal, as Antonio Faustino Carvalho has published. It's quite well represented in the archaeological record. It's not the most representative pottery, but is present in most of the archaeological assemblages. Until 2019, the limit of the expansion of this kind of ceramic production was more or less in Lisbon, so we didn't find any cardial pottery in the north. However, recently in Covairos, they discovered a quite nice cardial water that is represented in the slide. And we tried to choose all this pottery, the earliest cardial in the Atlantic coast to analyze. However, this earliest pottery in the south is not the same in the north, especially in Normandy and Brittany, where the earliest pottery is completely different. The decorative, the morphology, and of course the technology is quite different from the south. So we are speaking about different kind of pottery productions, different technological knowledge, and we wanted to explore if the use of this earliest pottery was exactly the same or not in these early farming societies. In France, especially in Normandy and Brittany, we have mainly two kinds of productions. We have the Lake Rouennais, Vicky Villeneuve Saint-Germain, that are characterized, or I always choose that photo because it's really nice, I have to say, from Berson. And after we have another kind of production, Lauguet pottery, that normally in the literature appear as pottery by hunter-gatherer groups. So we choose pottery from both sides, and we try to distinguish the use of this early pottery in the north of France. So the framework of our project was this. We choose a lot of different sites from the south of Portugal to Normandy. Was quite nice at the beginning, quite big at the end, a lot of work, but we tried to choose the well-established chronological and archaeological context, and we choose different archaeological sites characterized by different functionalities. We choose open-air sites, domestic areas, after funerary record, to see if we could distinguish different use of the early pottery in this site. So in this plot, we have plotted the results of our project. As you can see, its point is a pottery sample. So it's mainly the isotopic values of the main fatty acid, the C16 and the C18. So in this plot, you can see the comparison between the isotopic values of our pot with the reference values in our database. So we can distinguish family of foods. So in the area we analyze, we can see a quite strong correlation in the use from south to north. We can see that in the south of the Atlantic Europe, especially in Portugal and north of Spain, the early pottery was mainly used to process meat that we have correlated with the processing of domestic animals. However, if we move to the north of the Atlantic Europe, we can see an increase in the use of pottery related with the dairy products. And the use of this pottery related to the dairy products is really, really strong in England. So we can see this strong correlation in the isotopic values related with the increasing of processing of dairy products in the north of the Atlantic Europe. I think I like more this one because it's really, really visual. So you can see that from Portugal to North UK, the increasing of these dairy products in the early pottery is really, really, really strong. So in our study, we have correlated this importance of the dairy products with the archaeological record. So this, the different uses of the early pottery in the Atlantic coast of Europe is quite well correlated with archaeological remains. So in the south, the processing of meat in the early pottery can be correlated with the importance of ovica prints in the archaeological record. However, in the north, this increasing in the dairy products can be correlated with the more presence of cows in the archaeological record. So we have established two kinds of exploitation, or maybe two kinds of pottery use in this Atlantic coast, one in the north related with this kind of archaeological record, and in the south related with the ovica prints.
Okay, so thanks, Miriam. So that's a really good overview, and you can see how we, on a regional sense, we can start bringing big data sets together to look at how differences in pots are being used, how meat and milk has been used differently as you move through time and space. However, what we also wanted to do, and what we're really working towards now, is looking on a pot by pot basis and looking within a site to see how those different foods have been processed in different vessels. And obviously this requires a close link up with people actually studying the pottery itself, the vessel forms, the shapes, the volumes, and the technology as well. But one thing that we've been doing is looking at this idea of cuisine and asking this question, how food was actually being valued, how was it being processed, what choices were people making in the terms of the foods they were choosing to put in a pot. And so we might have a site where we have lots of foods available. So we have meat from ruminant animals, for example, we have porcine products, pork, we have fish potentially available, and we have dairy products such as cheese. But it's interesting to see which of those products actually ends up in the pot. And what's really nice, if we can think about whether, if we don't find one of those products, we're finding an emphasis on certain things and not others, then perhaps we're looking at some kind of taboo or exclusion or choices that people are making that's excluding certain foods. And very quickly, we've done a little bit of work on Miriam's data set to study this. And if we amalgamate the data and we actually look at the values of the isotopes present in the pots, and we look at the number of pots represented by each category, we start seeing some interesting patterns. So this is in southern England, for example, and we don't see, if you look at the distribution of the data, you can kind of make out three humps, three different distributions. And this is probably telling us that people are beginning to separate out pork products from ruminant meat products from dairy products. They're actually using pots differently and separating them for these different uses, which is really interesting. They're not just putting everything in the pot. If they did that, we should see an even distribution. In Portugal, for example, we see certain pots only being used for dairy, whereas meat, which is both ruminant meat, so sheep and goat and cattle, but also porcine, pork products being mixed together in the pots. So it looks as though they're separating out some pots just for processing meat and other pots just for processing some dairy products. Whereas in Cantabria, we found a much mixed signal, so less samples, so we need to define that, but perhaps more the pots have been having longer use lives and are having lots of different products mixed together. So that's really room for the future, and we need larger sample sets. We need to look site by site and to try and build up this data set and also combine it with the actual forms and shapes of the vessels. So, okay, what about plants? I think this is really exciting. I think this is the future of residue analysis. It's really challenging. What can we identify? And of course, we're looking at the fats mainly. So things like olive oil is a possibility to identify in pottery. It's actually quite difficult because a lot of plant oils have the same kind of molecular constituents, but we have ways of actually distinguishing these now. Probably not so important for the Neolithic, maybe towards the final part of the Neolithic and into the Bronze Age. It'd be interesting to try and identify whether we can identify some of these oils in certain vessels. A lot of work which is not relevant to us in the Mediterranean is on brassicas. These have got a lot of waxes in, they're very waxy plants, so these can be identified. Tree bark resins can be identified very easily as well, not really to do with cuisine, of course. But I guess the holy grail and what we really want to get to is to start to identify cereal production in pots. So a lot of focus on meat. We think we need to change, and we need to start to use different approaches to identify these. A quick example, this is a site from the Luschen Pass in Switzerland. It's an early Bronze Age wooden vessel with a residue on the inside. It was amorphous, you couldn't really say what it was. And we believed it was plant foods. And we did some analysis, and we were able to identify these new biomarkers, these chemicals that are actually from the cereals. They're called alkyl resorcanols. It's a long word, but we can able to use them and actually identify split things like wheat and rye and barley apart. So there's a really interesting way of approaching the cereal study, and we do have biomarkers for those. Whether they survive or not is a different question. There's been some work done at the Roman site of Vindolanda in north of England, which is a waterlogged site. And the thought is now that these molecules really survive in kind of waterlogged conditions. So thinking sites like La Mozota would be ideal for this kind of study. The other thing we're able to do with this approach is actually to pull out the proteins. So not the fats anymore, the actual protein part of the plants. It's mainly carbohydrates, which that doesn't survive, but we're able to pull the protein out. And with that, we're able to get much more specific identifications all the way down to families and species. So that was also applied to this, and we were able to narrow it down to certain types of bread wheat. So really excited for the future. So really to finish, what about alcohol? And we always get asked this question. There has been work done. There was studies that were kind of done in the 80s, I guess in the 90s, where they're using palynological approaches to look for beer production in pottery, particularly in beaker and Bronze Age pottery. You know, it's difficult to actually tie those palynological identifications to the brewing of beer and identify them directly to the pot. You know, people have said for wine, for example, which maybe is more relevant for us, really difficult. It's always had challenges. There's been a lot published. A lot of it is being contested in the literature. It's not really well believed. So if we look at what's the evidence for Neolithic wine, well, there was a study published a few years ago where they identified this marker. It's called tartaric acid. It's present at a high concentration in both red and white wine. And they found it in sites in the Caucasus, dated from around 6,000 to about 5,800 Cal BC. So this is one molecule. The problem with that is this molecule is present everywhere, and a lot of people are rather skeptical about that identification alone. I think a probably more secure identification comes from the site of the Kilitash, an Neolithic tell site in northern Greece, where not only did they have the tartaric acid in the vessels, but they also had the actual seeds present, and they looked to be crushed and processed to more suggestive of wine. So you have the botanical evidence and the chemical evidence together. More recently, though, we've had some advances in the kind of methodological approach, work carried out by Leah Dreer when she was doing a postdoc with us in York. And what she was working on Mediterranean transport amphora, but what she was able to do was much more securely derive criteria for the identification of wine, not just using the presence of this one molecule, but looking at the ratio of these two different molecules, which can really start to separate out wine from some of the other fruits. The problem with the tartaric acid is present in a whole range of different fruits. So but by using these different criteria, we could be more specific about the identification of wine. And we think this has got really exciting promise for the future. And what Leah wants to do, and what we want to do, is really start applying that to earlier vessels, going back through time into the Bronze Age, into the Neolithic. And so finally, just to say, we've talked a lot about cooking pots and cooking vessels, but we're also looking to expand residue analysis into different artifact types. So these amalgamated charred material, we saw some yesterday at La Mozota, potentially where we've got cereals identified in them, potentially that's a good target for doing residue analysis. What else is in those mixtures? Is it just cereals clumped together, or is it being cooked with other types of foods, animal products perhaps, can be identified? Pits, a lot of work has been done on that. So these are slabline pits from Norway, but other types of pits as well. Whether they're lined or not, we can identify the soils within the pits and try and look at kind of chemical composition of them to identify the products that are being processed within them, if that's the case. Some work on grinding stones. We don't think that lithics are going to be very useful for doing residue analysis, chemical residue analysis, because they've got a very non-porous surface. But perhaps grinding stones, where you've got more porosity within the material itself, and it's come into contact with a product for a much longer time, has potential for proteins, potentially DNA, but also for lipids as well. And then other kinds of things. So these are like cooking stones, object heated stones that are placed inside vessels or placed inside other types of containers, perhaps for heating, could also be a good target for residue analysis. So we've got a project at the moment with Clement, 15 PhD students, exploring these different types of materials.
Ho preso una strizza con quei due. Quella, vede uno e se lo fa nel...
Okay, so that's us. Thank you very much. Slab-lined pits from Norway, but other types of pits as well.
Eva?
Whether they're lined or not, we can identify the soils within the pits.
I'm here.
Look at chemical composition of them to identify the products that are being
I'm good, thank you. How are you?
Okay. Some work on grinding stones. We don't think that lithics are going to be very useful.
Yeah, I'm going.
So they've got a very non-porous surface.
Por favor, saca al niño que queremos.
Child, I'm afraid he's in nursery, so está en la guardería. Okay, so all right. Yeah, can you see my screen? I'm gonna make it like full screen now.
Yeah, okay.
Yeah. So here we are. Buenos días a todos. Quería agradecer a los organizadores la invitación. I would like to thank the organizers for allowing me to speak today. So in this talk, I'm going to try to summarize how ancient human DNA has contributed to the knowledge of the first Neolithic populations and to the spread of the Neolithic in Europe. Ancient DNA is a fast-moving field, and new data is being published at an amazing speed, and which makes it even difficult for us that we are in the field to follow sometimes. So if I have to give this talk in a year time, I will probably be telling a different story, because we will have more data and probably a more insightful overview. So before I start, I just wanted to make like a note saying that while most ancient DNA papers, I'm going to be talking about ancestries and about admixture. So while most ancient DNA papers, they will use labels such as farmers or hunter-gatherers, for accuracy, I will be using pre-Neolithic and Neolithic groups instead. As I understand, it could be confusing for experts in other areas, especially if we consider that there is not always a correspondence between the subsistence economy and the chronology and the ecological attributes of the individuals. So which type of questions has ancient DNA tried to tackle in the past 20 years? So we have a list here of things that ancient DNA has tried to address more or less successfully. So does the Neolithic package has a single or a multiple region of origin within the Near East? What is the relationship between the first Neolithic communities where they're preceding hunter-gatherer groups, Natufian or Epipaleolithic groups? How did the Neolithic expand into Europe? Was it a movement of people, a movement of ideas, or was it a mixed model of both? How was the interaction between farmers and hunter-gatherers in Europe? What is the main population source, if any, for the spread of the Neolithic into Europe? Was there a single population source? And did the spread of the Neolithic took place mainly through land via Anatolia or through seafaring directly from the Levant? So before we go into examining the different findings and the interpretations, so it is important to acknowledge the challenges that anyone working with ancient human Neolithic has to face. And the main challenge is always the lack of long-term DNA preservation in temperate environments. High temperatures, such as the ones that we find, for example, in the Near East and some southern regions of Europe, they promote the fragmentation of the DNA, and in some cases they wipe it out completely, making it impossible to analyze. And that in turn increases the risk of external contamination by people that have manipulated the samples at some point. However, two main technological breakthroughs have made possible, at least to some extent, to overcome this issue of degradation. So the first one has been the development of a new strategy of DNA sequencing, which is known as next generation sequencing or high throughput DNA sequencing or massive parallel DNA sequencing. So this approach allows the simultaneous recovery of short fragments of DNA. This technique was first introduced in the field of ancient DNA in 2006 to sequence mammoth DNA, and has replaced traditional sequencing methods very quickly in the study of human ancient DNA. And the second breakthrough was the discovery by Cristina Gamba and collaborators in 2014 that the DNA that was contained in a particular type of bone, the pars petrosa or petrous bone, in the temporal bone of the skull. This is the bone that surrounds the ear. This bone seems to be particularly resilient to environmental degradation, to the point that it's been measured experimentally that it provides between 4 to 16 times more endogenous ancient DNA than teeth, and between 100 and 200 times more DNA than any other bones in the skeleton. So these findings together, they have made possible to access genetic information from temperate areas such as the Near East and Africa. And at the same time, it has propitiated the hunting for petrous bones from osteological collections around the world, but this is a different story. So currently, genomic, so information from full genomes from different positions across the whole genome has been published for more than 500 individuals from around 150 locations across Europe and the Near East, spanning the Mesolithic Neolithic transition brackets between 1000 BCE to 5000 BCE. So this is a public information that is published in this site, and I've just I have like painted on top of the well, just to highlight the sites that they are Mesolithic enclaves here. So these numbers, they will get into the thousands if we include more recent chronologies within the Neolithic. So it's like quite a considerable body of data coming from ancient DNA. So what have we learned from ancient DNA regarding the transition to farming? So, well, one of the first things that we have learned is that the first Near Eastern farmers, they were already genetically diverse. So there are several studies that they have shown that the Near Eastern farming groups from the original farming centers in the Zagros Mountains, in the southern Levant, and also in Anatolia, were already genetically different, very different between them, and they were quite similar to the preceding epipaleolithic populations. And this suggests that they were mainly descendants from these epipaleolithic populations independently, and at least at the beginning with little input from other genetically distinct population groups. So we can see this in these two fancy plots. we like plots in genetics. So the one in the left is principal component analysis, and each of the dots that you have here represent a different individual with different origin and a different chronology. So explained in simple terms, this plot has two dimensions, has two axes here. And when we have like a series of dots that they cluster together in the same space next to each other in the plot, this indicates genetic similarities. And when they occupied distant positions in the plot, this means that these individuals, they are genetically different. So we can see how the first Neolithic farmers from the southern Levant, so in blue here, from the Zagros mountains and in Anatolia, they occupied distant positions in the plot, meaning that they were already genetically differentiated at the beginning of the Neolithic. And they are closer to their epipaleolithic counterparts. And what this means is that they probably descended from them with little input from genetic input from other populations. So the plot in the right, it's a different type of test. It's what we call a mixture test, and it's highlighting the same phenomenon. So you can see, for example, that the different colors, they represent different types of DNA. Like to explain it in simple terms, so you can see that the farmers from the Zagros here, they are genetically different, for example, from the farmers from the Levant, from the southern Levant. And they are in turn different from the farmers from Anatolia. These Anatolian farmers, there seems to be a mixture already of these Zagros farmers, these Levantine farmers, and they also have like a basal substrate from Western hunter-gatherers. So it was also noted in these first genomic papers that there was also a high degree of genetic structure among hunter-gatherers, which means that hunter-gatherers from different parts in Europe, they were also quite different genetically. So you can see here in the plot, for example, you can see three groups, sorry. Eastern European hunter-gatherers, Western European hunter-gatherers, and Scandinavian hunter-gatherers, they were quite different from each other. This study and also other studies, they have noticed also that most early Neolithic individuals from Europe, they have ancestry from Anatolian farmers. So these yellow dots that you have here, some of them you see that they overlap with the red dots corresponding to Anatolian early farmers. And this has been interpreted as a demographic input from the first farming groups in Anatolia into Europe. So individuals that they probably moved and from Anatolia into Europe. So this pattern, in principle, it argues for Anatolia as a main population source for both the Mediterranean wave of expansion of farmers and also the Central European Neolithic roots. And there are different papers that they have addressed these. And in the words of Matthias and collaborators 2018, they say that we confirm that Mediterranean populations represented in our study by individuals associated with the epicardial early Neolithic from Iberia are closely related to the Danubian populations represented by the LBK complex from Central Europe. So they seem to be very close genetically. However, is that the case for every single early Neolithic individual from Europe? It seems to be the rule, but there are some exceptions. So there's been a few early Neolithic individuals from different locations. For example, in this study here, they studied some Neolithic individuals from the Peloponnese, in Greece, from Frank Decades, and from Diros, And these individuals, they don't seem to fit, at least to derive from the same genetic pool as the other individuals. So they don't descend from these Anatolian expanding populations, and this suggests that other population sources in the Near East, they were also involved in at least a fraction of the spread, genomic spread of Neolithic genes out of the Near East into Southern Europe. But unfortunately, the source of this population movement is still unknown. This pattern has also been seen in another publication on several individuals from northeast Italy. So in 2014, we put forward the hypothesis of an early PPMB expansion directly from the Levant into Cyprus and the Aegean, and that was based on short partial mitochondrial DNA sequences. So while the data was limited, obviously didn't have the resolution and the power of all these new genomic studies, it offers an interesting alternative that needs to be tested to explain the origins of these individuals, that they don't fit the model of spread from Neolithic Anatolia. So maritime spread of first farmers from the Levant into Cyprus fits well with the archaeological evidence, and also with the evidence of early navigation during the Neolithic, from which we will hear more in some of the talks of this seminar series. So another thing that we have just begun to understand is that the Neolithic transition in Europe, yes, it involved a movement of Near Eastern genes westwards, but also that there were different degrees of admixture of these farmers with local Mesolithic groups. And there is also a huge body of data on this. Here you have data from two papers from Ribolad and collaborators 2020, and from Lipson and collaborators in 2017. So in the plot on the left, the x-axis represents time in years BCE, so from oldest to more recent, and the y-axis represents the proportion of pre-Neolithic or so-called hunter-gatherer ancestry in these populations. So while the table that you have on the right attempts to quantify the percentage of hunter-gatherer ancestry in three different populations, transects from the early Neolithic up to the Chalcolithic, in the Danube region in Central Europe associated to the LBK cultural complex, and also in Iberia. So you can see in both cases that there is a steady increase in pre-Neolithic ancestry. So the amount of hunter-gatherer ancestry or pre-Neolithic ancestry that was present at the beginning, it's different between populations. So we can see that, for example, in some cases, this admixture seems to have happened immediately and appeared after the first communities of each, farming communities of each regions. And in other places, it took much later to appear and was sustained for more than 1,000 years. So we see, for example, that in Hungary and in Central Europe and also in Iberia, the early farmers, they had like a small proportion of hunter-gatherer ancestry or pre-Neolithic ancestry, and this ancestry increases as the time passes, while in other populations, like for example in the Iron Gates in southeastern Europe, and also in some sites in south of France that they are associated with impresso-cardial pottery complex, we see significant proportion of hunter-gatherer or pre-Neolithic ancestry among the first farmers that they reach and they arrive to these regions. The cardio-impresa cultural complex is particularly interesting because we see this hunter-gatherer ancestry in early farmers, but we don't see the same pattern in the same cultural complex in the Adriatic region. And this is suggesting that probably this was a recent admixture event that happened on site with hunter-gatherers that were in the vicinity. The case of the iron gates deserves also further attention, as a combined study of ancient DNA and isotopes of mobility has helped understanding how these populations interacted between them. So in this case, when the first farmers moved into the region, they encountered a highly dense semi-sedentary Mesolithic population that had an economy that was mainly based on fishing. And in this case, the genetic evidence shows first that these Mesolithic individuals, they were already interacting with Anatolian groups. And we see that because these hunter-gatherers from the Iron Gates, they already display high degree of genetic affinity with Anatolians. So also they carry mitochondrial haplogroups that are typical of early farmers, like K1, for example. And there are few individuals that they display 100% Anatolian genetic ancestry. And the analysis of isotopes of mobility, according to strontium values, they were identified as probably being first-generation migrants, so individuals that they come from Anatolia. And other individuals from the sites, they display also high amount of this pre-Neolithic ancestry. So these mixed ancestry patterns, admixture between Neolithic populations coming from the Near East and a local pre-Neolithic ancestry is also observed among these early Neolithic individuals from the same region. So some individuals show a high proportion of this pre-Neolithic ancestry. And there is also a case of one individual belonging to the Koros culture, archaeological culture, whose genome, despite being in a Neolithic context, this looks like entirely 100% pre-Neolithic. And this is signaling phenomenon of acculturation, local individual that adopted farming. So to sum up, what have we learned in all these years of ancient DNA research? You have a summary of the main conclusions here. The first farmers in the Near East were already genetically diverse. The majority of early Neolithic farmers from Europe, they have ancestry from Anatolian Neolithic farmers, and this can be interpreted as a demographic input from the first farming groups, mainly from this region. But we also saw that there are other individuals that don't fit exactly this model. Pre-Neolithic hunter-gatherer populations in Europe were also diverse, and they were genetically different from early Neolithic populations from the Near East. And there was a spread of genes, but also admixture with local Mesolithic groups in different regions of Europe. And this admixture, in some cases, didn't happen immediately after the contact, and it took more than 1,000 years to complete. So, right. So just to conclude, is that all? So, well, obviously it's not. There's still some questions that still remain that haven't been answered, like, for example, whether there were other genetically distinct unsampled populations in the Near East, and whether or not they had an impact over the genetic pool of the first early Neolithic individuals from Europe. And in which form did this pre-Neolithic substrate survive, in some cases thousands of years after the, well, hundreds of years after the arrival of the first Neolithic groups, and how this increase that we see in some regions of pre-Neolithic ancestry happened. Can ancient DNA also help in researching the social consequence of the adoption of farming, maybe by looking more at individuals, site, and biological relationship between individuals? So I believe that the answer is not just in ancient DNA, and that there is a need currently that we are holding like a sufficient amount of data to like zoom in, and this is already happening at site level, family level, individual level, and engage more effectively with archaeology, anthropology, ethnography, and sociology to provide like a nuance, a more comprehensive answer to all these questions. And we need to remember that ultimately what we are trying is to reconstruct the past, and ancient DNA, like any other discipline, is subject to errors and misinterpretations. So that's all from me. So thank you very much for your attention, and I'm happy to answer any questions you may have.
Hi, thank you, Eva. Thank you for your talk. It was really interesting. So I don't know if we have any questions on YouTube. No. Okay. So yeah. Okay, perfect. So we will keep the questions for the end of the seminar. So we have now the coffee break. So we will come back at 11:30, I think. Yeah. So thank you, everyone. So we will take the coffee. Thank you, Eva. Amy Bogar. Hello. Your microphone.
Hello.
Hello, how are you?
Good morning. Hi. Good morning, everyone.
If you want
to say hello?
We can try your presentation first.
Of course. Shall I try to share my screen?
No, we don't have.
Yeah, it's okay.
It's okay?
It's okay, it's okay. Well, 11:30 we come back.
Yes.
We take a coffee now. So. Debemos salir fuera porque no puedo tomar café aquí por cuestiones sanitarias.
Successo, un bel coraggio.
Auf Gleis 3! Schnell, schnell! Kommt, kommt!
Successo, un bel coraggio.
Okay. Okay, Amy.
Thank you. Let me share my screen. Can you see that? Is that clear?
You open the presentation because we see the slide in the left.
Ah, okay. Let me try again. Maybe I just share my whole screen and then I will take away everything else. Is that better?
No, yes, okay.
Great. Thank you very much. And good morning, everyone. I'm sorry that I'm not there drinking coffee with you in Rome, which I would much prefer. But it's already been a wonderful morning, and I'm really grateful to the organizers for the opportunity to participate. I'm going to speak about Neolithic farming in Southeastern Europe. And I want to start with the very general observation that we have unusually good conditions for the investigation of early farming and its social context in this region. And I would just like to draw your attention to some of the reasons why those conditions are particularly good. So partly it's that we have these different forms of site to play with. And of course, there are other forms of site as well, caves and so on. But I focused on those that are of particular relevance to some of the results I want to present today. So of course, the classic tell sites, the settlement mounds that often feature, for example, burned houses, burned buildings that have intact in them assemblages of crops and other material that were being stored and prepared in the house. So that's one kind of scenario. We also have in Southeastern Europe flat extended sites which do not form tells, which drift horizontally, but which have yielded some really spectacular evidence for large-scale animal consumption and give a sort of complementary window onto the scale, even the regional scale of some of those feasting events. And then, of course, we have a wetland Neolithic. We have lakeshore sites, and I hope that we'll hear a bit more about some of these in other talks today. The lakeshore Neolithic of a particular region of Southeastern Europe, the map that you can see in the lower right-hand corner, is the focus of an ongoing ERC synergy project called EXPLO. This is a collaboration between the universities of Bern, Aristotle University of Thessaloniki, featuring Kostas Kotsakis, who will join us later today, and myself at the University of Oxford. And I'll draw on some of the emerging results of that project to try and illustrate the opportunities, complementary opportunities for looking at Neolithic farming from a wetland, waterlogged perspective. So those are one set of reasons why Southeast Europe is interesting, but I note, of course, that the central and western Mediterranean regions of Europe, which we're hearing so much about today, also offer a lot of this complementarity. There are also some particular features of the material culture of Southeastern Europe that are just really compelling and evocative for thinking about the handling of food. Of course, the ceramics, which we've heard about already, but also other aspects of the clay repertoire, including miniature clay representations or models of houses. And I'm showing you here some late Neolithic, specifically Greek examples. In the late Neolithic, in the Greek house model corpus, there is a particular focus on certain features of the house, including the interior of the house, which you see represented in the two examples in the upper row here, where we have miniature domed ovens within the rooms that are being depicted. And in the upper right example, interestingly, that room that's being depicted is raised. and it's been suggested that that's actually a pile dwelling in that case. We have another raised situation in the lower left-hand corner. This is from Tumba Kremastis, so this is in Western Greek Macedonia, as opposed to some of the Eastern Greek Macedonian examples shown here. And in the case of this model, the roof could be removed, and the idea is that figures and other items could be moved around and displayed in the house model. And then in the lower right-hand corner, this is an example from a site, Promachon, which is on the border of Greece and Bulgaria, so Eastern Greek Macedonia. And in this case, the heads of animals would appear to be bucrania, so the heads of cattle are shown hung on the external house wall, either side of the house entrance. So just very rich, evocative representations of the house's interest in the handling, the preparation, and also the display of food-related items. So against that kind of rich background, these are the aims of what I'd like to cover today. And a big theme already this morning has been the diversity of practice. And that's a theme I hope to speak to as well. So the diversity and dynamism of Neolithic farming. It isn't a simple cartoonish understanding that we're getting now. I think it's much more detailed and site-specific and regionally variable. And I'd like to give some examples of that. But alongside that diversity, there is also a convergence. There is also a kind of basic theme that reemerges in various case studies around particular approaches to cultivation and herding that I'll point out as we go along. And then the other thing I'd like to try to keep pointing out is that the complementarity in opportunities that we have for investigating early farming from these variously dryland and wetland Neolithics. I'm actually going to start with Knossos, one of the best known Neolithic and also Bronze Age sites in Europe, of course, but focusing here on its Neolithic phases. Knossos is one of a set of sites, some of which you see in the upper right-hand corner map, which date to the very initial expansion of farming around the Aegean. The Neolithic sequence at Knossos is pretty remarkable, though, and unique in that it continues from that initial phase right the way through to the final Neolithic, so a period of almost 3,000 years, and then, of course, famously into the Bronze Age, culminating in the formation of the palatial center of Knossos. The Neolithic settlement here starts off very small. It's about 0.3 of a hectare is the estimate for the earliest aceramic Neolithic phase. It then expands in the late and final Neolithic to a couple of hectares or perhaps as much as 4.5 hectares. And I want to talk about the effect that has on farming shortly. And then in the lower right-hand corner, I'm just indicating some of the evidence that we have for domestic architecture, which is increasingly sort of substantial and reflective of discrete households as we move through the Neolithic sequence. And that process is underlined by the occurrence also at Knossos, as is better known from northern Greece, of clay house models. And there's a fragment of one example that's been published by Peter Tompkins that I'm showing here. In the far left, I'm just including here a Google Earth shot of the site of Knossos. The Neolithic site is under the Bronze Age palace along the Kairos River Valley, which is still a focus of market gardening and intensive cultivation today. Now, Knossos is one of the case studies that we've been looking at in an ERC project called AgriCurve, which is officially finished, but we're still publishing from it. And this is collaborative work with a number of others that I will acknowledge at the end of the talk. What I'm showing you here is some of the results of an integrated plant and faunal isotope study through the Neolithic and Bronze Age sequence. We have much better coverage of all of the Neolithic and Bronze Age phases in terms of faunal data. And we're showing you the sheep nitrogen values here. That's the right-hand graph for reasons that I'll explain in a moment. Well, sheep is one of the dominant, of course, constituents of the faunal assemblage, and it is the dominant domesticate through the Neolithic period. Then on the left-hand side, we're showing the results of stable nitrogen isotope analysis of very well-preserved crop material that we select carefully. It's within a certain preservation window where we can control for the effects of charring, for example, on the isotope values. So in all cases here, I'm showing nitrogen, and I've picked out sheep to compare with crops for one particular reason, and that is to draw your attention to the way in which the sheep and cereal and pulse nitrogen isotope values appear to track each other. So they start relatively high, they shift downwards as we get into the late Neolithic, they bounce back up as we get to the, by the final Neolithic, which is represented also in the archibotanical assemblages. And then they start to go down again as we go into the Bronze Age. That's what I'm showing with this dotted line arrow. And that's a story that's beyond the scope of what I'm going to try and cover today. So I'd just like to talk about this Neolithic bounce in the carbon, or in the nitrogen, sorry, in the nitrogen isotope values. What's particularly interesting is that the sheep and the crops are co-varying, that they're tracking each other. And we think this reflects their integrated management in the landscape, with sheep largely grazing on cultivated land and stubble. And this is something that we've modeled in some of the graphs that you can see here. So the one in the lower right-hand corner, those ellipses are the colored ellipses are what we would expect for sheep that are consuming different kinds of diet of cultivated and wild habitats that are relevant at this kind of rainfall zone and based in part on the archibotanical carbon and nitrogen data. And the sheep ellipse, which is just the blue line that you can see, maps quite nicely onto cereal and pulse chaff and stubble. And so quite a lot of their food is, their forage is plausibly coming from that kind of habitat. And so whatever is happening with the crops nitrogen-wise is carrying over into the sheep. But why do the nitrogen values go down and then up again? Well, the most plausible interpretation that we've come up with is that the initial reduction in nitrogen has to do with the expansion of the settlement. We know ethnographically that cultivators face sort of frictions of distance the further that they have to travel to their cultivation plots, and that the intensity of management tends to decrease as settlements get bigger. If settlement density is high and cultivated plots are outside the settlement itself, which is what you would expect for a tell settlement like that at Knossos. So we think this initial decrease in nitrogen is those frictions of distance really kicking in as the settlement expands and as intensive practices like manuring become less frequent and less intensive. The increase then in nitrogen values in the final Neolithic coincides with an increased use of cattle for traction, but not the oxen, which become the classic sort of traction animals of the Bronze Age. These are cows. So these are animals that of course are kept for breeding, probably for milk production, dairy production as well. But in their final years are being used to pull a plow, to haul manure around the landscape, to bring crops in and so on. And that then eases those frictions of distance and again increases the scope of intensive gardening, of intensive cultivation. And I'm building very much here on the work of the lead author of the study that we're in the process of writing up, Valacia Izakidou, who's been working on this faunal assemblage and identifying increasing indications of pathologies on the lower limb bones and pelvis of female cattle, of cows, as you move into the final Neolithic. Before I leave Knossos, because I'd like to look at other case studies to compare and contrast, I want to show you these results briefly as well. This is the same crop isotope data that you've seen before, but now with carbon on the x-axis and nitrogen on the y-axis. And what I'd like to draw your attention to here is the fact that the pulse crops, the ones that are shown in the purple triangles and the purple triangles are below and to the left in carbon terms of where the cereals are sitting. So the cereals, which are blue and red, are above and to the right of the pulses, which are sitting at the same place carbon-wise or a little bit lower. That kind of relation between pulses and cereals is exactly what we expect if they're being grown under similarly well-watered conditions. We know that from experiments that we've conducted in Aleppo and also in more controlled conditions, and that is that pulses are quite sensitive to water input. And if pulses and cereals are being grown together, the pulses show even greater carbon values, they tend to, than the cereals because of their greater sensitivity in carbon values to their water status. That relation completely changes by the time we get to the late Bronze Age, this final palatial period, which is the lower right-hand corner. So we're skipping now to the later second millennium BC. And by that point, we have a different set of pulse crops. Now they're these brown X's and gray triangles, but they are now below and to the right of the cereals. They're grown under drier conditions than the cereals and almost certainly separately. So the contrast I'm trying to draw out here is between a Neolithic in which cereals and pulses are being grown either together through intercropping or by rotation, and a Bronze Age, a late Bronze Age scenario at palatial Knossos, where pulses and cereal production is now completely separate in the landscape. Pulses probably grown as allotment garden plots and cereals much more extensively elsewhere. So how typical is Knossos? Well, in one sense, every site is different, but as I've already anticipated, there are some themes that we can see variations on at different sites. So I'm showing you here crop isotope values, again, nitrogen for a couple of other sites. These are middle and late Neolithic sites, Halai in central Greece, opposite Evia on the coast, and Koufouvenou near Sparta in the southern Peloponnese. And in each case, something slightly different is going on in terms of how farmers invest manuring in different crops. So at Koufouvenou, if you look at the sort of second column of points, Middle Neolithic Koufouvenou, which is the second column of points over from the left. I don't know if you can see my arrow, so I won't assume that you can. But in that case, the free threshing wheat is particularly the crop in which manuring is being invested. It has relatively high nitrogen values, higher than those of barley, which are shown in red, and much higher than those of einkorn, which is another wheat crop that appears in the late Neolithic at Koufouvenou and seems to get no manure at all. So different niches, different growing conditions are being created for different crops, and this must have to do also with their culinary importance and role. At Halai, by contrast, all of the crops, all of the cereals and indeed the pulses are very variable in their nitrogen values. The pulses and cereals kind of track each other, so they're probably grown in an integrated way, but there isn't any particular crop that is privileged in terms of its growing conditions. So it's a different approach again. Just before we leave these examples, I wanted to point out that for Halai, we also have human data as well as good faunal isotope data, as indeed we have good faunal isotope data for Koufouvenou as well, but I'm just being selective here. In the case of Halai, what's nice is that we can see then in paleodietary terms how these different sources feed or don't feed much into the human diet. And in a nutshell, it appears that the humans, which you see in black symbols towards the top, along with some dogs and some pigs, as you'd expect, they are plausibly a trophic offset in nitrogen and carbon terms away from the very variable in nitrogen terms crops that you see lower left. So this is an opportunity to test the assumption that we otherwise make, which is that this set of crops is in fact the foundation of the diet and of subsistence. Much less so the fauna, and certainly not the cattle, which you can see are enriched in carbon. They're probably nibbling on C4 plants in marshy habitats along the coast. And that's a phenomenon, interestingly, that we pick up at other coastal sites in Greece as well. So we've talked a little bit about insights that we can gain from faunal remains, especially in this case through isotope analysis, but also pathological analysis, and through isotopic analysis of charred plant assemblages. most usefully preserved in their original storage context in houses. There are other insights, as I mentioned, that are available from flat extended sites, especially around animal consumption, but I won't take time here to elaborate on that. Instead, I'd like to use the remaining moments that I have to look at a lakeshore example that is coming out of the Explo work and that we've been able to continue work on through the pandemic. I did want to acknowledge the sign for the dig house at Dispilio on Lake Castoria, which is at the very base, the very southern end of the map, the Google Earth map in the lower right, where Kostas Kotsakis, my collaborator on the Explo project, has just finished his excavation season, and in due course we will look forward to presenting results from that investigation. But in fact, the lakeshore example that I want to zoom in on is one on the southeastern shore of Lake Ohrid in the Republic of Northern Macedonia. And I'm delighted that Goce Naumov is here too, and will speak to us a little bit later, and very much want to acknowledge his support in this work. Ploča is interesting in part because of its location. It's in a transitional zone climatically and biogeographically. It's on a series of historic communication routes, and it is a submerged late Neolithic site that gives us an opportunity to consider the kinds of bioarchaeological ways into farming practice that are enabled by waterlogging. I want to give a shout out now to Amy Holguin, who is a PhD student here and has been working on Ploča as part of her PhD project since last September, and also to the other collaborators on Ploča whose work I'm drawing upon here, and I'll include everyone in the acknowledgement slide at the end. But, and in fact, it's to Amy Holguin that I want to particularly thank for the sequence of slides I'm about to show you. This is an aerial shot that shows you the position of Ploča and of the modern museum and of the reconstructed platform and dwellings that are there for visitors to see. The platform that you see in that white rectangle is partly overlying the submerged archaeological site that I think you can sort of make out the extent of on this kind of shelf on the edge of the very, very deep lake of Ohrid. So one of the ways we investigated at this site in 2019 was through a coring program, and this was made possible by the coring equipment and the raft and coring station of the paleoecology team led by Willie Tinner, who's one of the PIs on the ERC Synergy Explo project. And the coring team then did sort of crosswise transects of the settlement area. And it's the cores that are marked in red on the right-hand side that are the particular focus of the work we're doing at the moment in the Archaeobotany lab here in Oxford. And you can see some schematic profiles of those cores on the left-hand side. What was unusual and very interesting about Ploča, partly because it's sitting on that shelf at the edge of a very, very deep lake, is that you get a steady accumulation, almost a midden-like accumulation of organic debris, a meter and a half, even 1.7 meters thick in places, and seemingly continuously deposited, so there aren't any sterile layers in this sequence. So it's giving us a great big wodge of organic cultural material to see what people are doing food-wise through time. The chronology of this is quite tight. It's a period of maximally about 150 years, according to the top and bottom radiocarbon dates that we have so far. We're going to be able to refine that further thanks to the dendrochronological work of the team of Albert Hafner at the University of Bern in terms of felling dates. We may be able to get this down to just a set of decades that is represented by this cultural layer. And it's particularly this core very near the modern museum reconstruction platform that we have been working on and that we are going to present to you now. So one of the shocks of working on waterlogged material is the abundance. So we are routinely finding hundreds, even thousands of items per liter, meaning in any teaspoon or tablespoon of this stuff, you could easily spend a day analyzing what is there. This is just in terms of plant macro remains. And we've picked out here the particular samples in this core sequence, this core 0, where we find over 10,000 items per liter. The reason we've done that is that is a threshold that has been used in other wetland Neolithic areas, especially the alpine foreland lakeshore sites, as a kind of indicator of super good organic preservation. So we do hit that level periodically through the core. Now, this is just an overview of the kinds of plant remains that we find. And one of the big headlines here is that because we have waterlogged preservation, we don't see the same kind of bias towards crop material that we normally have on dry sites. On dry sites, where all plant material has to be carbonized in order to be preserved normally, we definitely see a bias towards the plants that are being stored and processed in proximity to fire throughout the year. Here instead, in this waterlogged assemblage, where there is some charred material, but most of it's waterlogged, what we're seeing is a much more even balance between crops and other foraged plants. So the red category, the fruits and nuts, which feature prominently in all of the samples and even dominate some, that's what we're routinely missing from charred or carbonized assemblages. And here are some examples of some of the fruits and nuts that we find, and I know I need to speed up a little bit. But the point here is to really give you an idea that we get this, technicolor diversity of really what the plant spectrum is that these communities are using, including things that are not that well preserved by charring normally, oilseed crops like opium poppy and flax as well. But back to this overview, and despite the lack of that bias towards crops, it is still true that many of these samples are dominated by crop material. And I just want to give you an idea of what that stuff looks like. We get the waterlogged and charred versions of the same thing. A lot of it is the discard of chaff from cereal processing. So that was clearly a routine activity. And also from chaff processing. So we get the pulses, the testa, and also the pod of pulses in both charred and waterlogged form. But the really dominant crop component that we find is from the dehusking, that is the final removal of chaff from the grain of what are called gloom wheats, so wheats like einkorn and emmer. And we even get an idea of the particular way in which this was removed, because we find this stuff mostly waterlogged, a little bit of it is charred, and then you get this kind of sweet spot in between where the chaff is singed or toasted, but not fully charred. And that will be of great interest, for example, from an ancient DNA perspective. Some ethnographic work actually in the Asturias region has shown how some of this singeing and charring takes place. It's plausibly from the removal of awns, the bristles on the cereal ear before dehusking happens, because the awns are a real pain. They sort of get in the way of just pounding the spikelets and getting the grain out. So probably a lot of the charred assemblages we have that aren't from storage contexts are from this kind of routine practice. Final thing I want to show you, and I will wrap up then, quite a bit of the assemblage, the green stuff that you see in the far right hand of these bar charts, are what we're calling here wild or weed seeds. And one of the big question marks with waterlogged assemblages is always, is this arable weed material, is it coming in with crops? Are these, wildflowers and other species that we're growing with crops in crop fields, or are they coming in through some other route? And fortunately, we have a means of testing the hypothesis that these are crop weeds. And this is from the work of Glynis Jones. It's a kind of approach that we also apply on dryland sites to try to understand the weed flora, because it gives us such valuable ecological information about crop growing conditions. And to make a long story short, we need to look at the aerodynamic properties of the weeds, because they behave differently in winnowing and coarse and fine sieving in order to tell archaeologically what kind of crop processing stage we're looking at, or if indeed we're looking at crop processing at all. In the case of plotcha, where we've included it in this kind of analysis, this is actually a discriminant analysis based on modern ethnobotanical study of the crop processing sequence by Glynis. And what we see is that, in fact, the plotcha material mostly looks like fine sieve byproduct. That's exactly what we would expect on the basis of the dominance of gloom bases. The gloom basis being what is generated by the dehusking of gloom wheats like einkorn and emmer. So this in fact confirms or supports the hypothesis that those wild weed seeds are in fact arable weeds. That then gives us kind of license to look ecologically at that weed flora about what it implies for growing conditions. So we haven't been able to take an isotopic approach here. We don't have much charred grain, but we can look at the weed flora. And this is a discriminant function that is a model based on a series of modern studies in and around the Mediterranean, where we've looked at what we're calling high input and low input agriculture. High input basically involving manuring, lots of weeding, lots of close attention to the crops. Low input being much more large scale, extensive type of approach. And this is all based on what we call functional weed ecology. So it's different traits that weeds have that enable them to do well under different kinds of growing conditions. And so we look at those traits in modern specimens, and then we measure them for modern specimens that correlate archaeobotanically with the weeds that we're finding. And that's what we've done for Plocha. So this is now what we get when we plug the Plocha samples into that discriminant function as unknown cases. We see a lot of variability, which is already interesting and I think plausible, because that's what real farming systems look like. But most of them align with the kind of lower end of the high input systems. And that's very interesting. It's very similar to some emerging results we have from other parts of Southeast Europe, and indeed going all the way back to Çatalhöyük in the Anatolian Neolithic. It's very similar to that kind of approach to small scale, high input farming. So just quickly then, some conclusions. It's increasingly clear from our data that farming practice in the Southeast European Neolithic was dynamic, was variable. There is, though, convergence around a small scale intensive approach, which I think is fundamental for appreciating socially and ecologically what the consequences of the spread of farming were. And finally, that the dryland and wetland Neolithics give us different windows onto the adaptation of this small scale intensive approach to different landscape settings. And I just want to finish by thanking the project teams involved, and especially my co-authors on some of the unpublished work that I've shown. Thank you very much.
Thank you, Amy, for your talk. So if you can keep a little bit, we will have the questions at the end of the morning. So we have our last talk. So Thomas. Do you need help?
At the mouse. Okay.
Yes.
Okay. I would like first to thank the organizers for the invitation, and also the host institution for giving us the place to meet each other. So we will present. I'm not sure. I know we will talk. That's why. Okay. That's fine. So we will present the okay. We will present the database and interactive web app for radiocarbon dates. And to work on this database and app, we are I, Thomas, and Nicolo Mazzucco, Miriam Cubas, Juan Francisco Gibaja, et Xavi Homs. So, the presentation, as you will see, was mostly based on ITs, not very much on the scientific or the archaeological purpose. So, we will present the database and the app and IT settings around this. At first, starting with the database. The database is basically composed by three files: the dataset, the material life correspondences, and the references of the dates. So the two first one are TSV extension, it means tab separated values, while the third one is the bib text. And as you might know, the bib text is a plain text used for bibliographical references. So actually, currently the database counts 204,052 dates coming from 1,752 different contexts, and this context belong to 542 different sites. So the interest of the TSV file is also on the GitHub repository. The layout is already updated. Looking for the chronological distribution of these radiocarbon dates, we've decided to constrain the period to select only those between 8,000 and 5,000 Ncal BP. And afterward, we calibrate all the dates, the dates with the Ncal last reference sheet. The geographical extension is the north, central, and western Mediterranean watershed. We thought it was a good idea to not work by country or by rectangular area, but mostly by watershed, since watershed are relevant geographical entities. So the application is a R-Shiny interactive one, and it's mostly like this, a distribution of points which can be gathered into, can be grouped into polygons at different size. I'm sorry for the quality, but I will show you a live demo afterwards. The missing data in our data set, the context, there are 7% of the context missing, and the type of material, 8%. The context is the column fast code, and the material is a material and material species as a specification of the material. Now, looking for the material, we made the correspondences between the type of material, which can be a lot of different things. It's case sensitive, so we have some kind of duplicated, and we classified this material in 3 categories of life duration: long life, short life, and other ones. Looking for the references are all in bib text. You might know this format. It's the open standard format for bibliographic references. And once transformed with our process, we have got something more readable. Okay, I will pass to the demo. Just skip for a second. Here. So actually the database, no, the app is on internet. I try to copy and paste the URL. It's not working. There's this little icon, which is bad. I think we are. Actually, it's on a Shiny app, server, open source server, but we are pushing the app on the University of Pisa. So, as you see, this is all the data set we have, and it counts, okay, that number, it's 2,446 datings. It's cover all this area, and Nicolo is calling. Sorry, I will respond. Okay, Nico. Nico, ¿me entiendes? Hola, Nico, ¿tú me entends? No, he was supposed to explain the archaeological purpose, and I was supposed about the 80. So each color refer to how can I hide this? Each color refer to a period, and you can select by different type of period. For example, let's select what interests us, the late Mesolithic and the early Neolithic. And you can choose the map background, and there's, I don't know where there's two, there's a gap, the glitch with these two one color. You can select also by Calvici, but it doesn't seem to work also. Okay, there's some glitch when you're pushing on the internet, but after each date has its own information and URL link when there exists a DOI, and you can also select a zone inside, and only this site will be selected. It's useful to sample, and finally you can select by the material duration, lifetime duration. So I go fast, and maybe there's not everything's working as it should be, but it's a matter of few things. The interest also of the application is when you have a number of you can select one date and or various dates and make their calibration on the fly. For that, I am reloading the NeoNet because there's this kind of glitch. I hope internet will follow. Okay. Once again. Okay, let's go to the Margineda, for example. Here is the Margineda. It's about 13 dates. Here we see we have 4 sites and 22 dates. So we want only this ones. Just see. Okay, 1 site and 13 dates. It's correct. Then I choose one. I have choose one. I go to the calibration part, and it will on the fly make the calibration of the 13 dates. And afterward, I can group by fast code. It's mean by context, or by site. There's only one site, silent period, or by period. And I guess it's only early Neolithic and all the dates. And here you got all the data, and there the bibliographical references, and here the credits. Okay, I will follow now the presentation with the presentation. Okay, here we are. So there's also a website, and the website is more or less the tutorial of the application. So the application was created with R, which is the most common used programming language in archaeology, and only here on your left you will see all the software used, the most used software in archaeology when they are quoted by the author. So ours largely come at first, and then Python, and then QGIS, and then mod apps, and so on. And R also allows to create packages, and these packages can share function, or can back up one with another, and so you can construct a real ecosystem of software. Here I present three packages, R packages developed for radiocarbon dating. The app, the NeoNet app, was also developed on GitHub. GitHub is hosts 190 billions of repositories, and is used by more than 40 million of people. It's a big platform allowing internet hosting, software development, versioning of dataset, of code, and so on. So continuous integrations. That means that there's a continuity between the code development and it's checking, license managing, contribution managing, issue threats, so and so. And the system, the logic of GitHub, it's a push and pull exchange between a standalone laptop and internet or cloudy repository, and it allows contributions with a system of Git, which means you can duplicate a repository, or you can create a branch on the main repository and then merge the branch with the tree with the main repository. The app we've developed is also coherent and follows the fair recommendation of the open science. it means that we make the information, the data information, findable, accessible, interoperable, and reusable. And our strategy was to publish the data set as to get a DOI, digital unique identifier. And after to publish this DOI in the Journal of Open Archaeological Data, with this title, NeoNet dataset, radiocarbon dates from late Mesolithic, early Neolithic transition in the north central western Mediterranean watershed. Also, the package C14 Bazar package developed among others by Clement Schmidt as a series of getter functions. These functions allows to map and to extract the information from more than 20 database, radiocarbon database. Maybe you will recognize the EuroEvol, the famous EuroEvol database from Manning and Shenan, among others, or maybe the Jomon one, developed by Enrico Crema and others. And I have contributed to get the BDA published by Perrin and Manin. And of course, I will create the function to read the data set and to make it readable by the C14 Bazar package. So here is come the part of let's say more historical cultural part. And unfortunately, as you've seen, Nicolo is working on the field, and it seems unable to connect. Juan, can we just check if Nicolo is on the connected, or should I? Maybe he's here. Yeah, un montón de mensajes. Millán. No está. Okay. Okay. So I'm really sorry, but I will be too imprecise to comment the following slide, so I won't. But it's an application. Nicolo Mazzucco used the application, and of course his knowledge about the context to to map and to extract some relevant patterns. I cannot. Riprendi la.
Is it possible? Yeah.
Here we are. So I will not comment anything, just let you some.
Prima di sganciarci dall'orbita dovremmo comprare da mangiare, avremo fame, siamo a 200 milioni di anni luce dal nostro pianeta e non c'è un solo buon ristorante lungo la strada. Odio fare la spesa.
E una birra, bella fresca, eh? Consumatori che vogliono la bella sì, se bella fresca, eh? Da sempre.
Okay. It was a nice silent presentation, but I'm coming back with my full accent. Okay, next development. So we plan to yes, we plan to change the watershed and to look at the west. And so it will be the same principle. We hope that IT and the collaborative work will be easier. We made some mistakes about how to share the data, how to update lines, columns, how to fix the column names, and so on. So I guess the next data set will be easier to look at and to integrate. And special interest maybe for me, it's to include Bayesian modeling with app. So, for example, we have filled the fast code, the context column, and we have unified almost the stratigraphic layers, and we also add for each layer we have chronological dates, absolute dates. So it should be possible to include a module, a function in the web app, allowing to perform Bayesian modeling, since also chrono model exists, is integrated into R. Thank you for your attention, and sorry for the different glitches.
So now we have some time for yes, please, if you open your camera, we have time now for the discussion. So if we have any questions, suggestions, comments, or anything, now is the moment. So I have been checking the chat in the YouTube channel, and we have some questions. So we have the first one is for Carmine. So Juan asked, how do you perceive the contact between the Mesolithic and the Neolithic communities in southern Italy? Yeah, sorry. Yeah. The question is, Juan asks, how do you perceive the contact between the Mesolithic and Neolithic communities in southern Italy? I'm sorry, you have to come here.
Je vais répondre en français. Okay. Donc, c'est pas possible de parler de contact, mais nous, on a parlé de continuité entre les deux systèmes techniques, le système technique méso-castelnovien de septième millénaire et le système technique de tout premier néolithique. Donc, à ce moment, au niveau des analyses technologiques, on a les éléments pour parler d'une continuité entre le 7e et le 6e millénaire au niveau des systèmes techniques et au niveau des transferts des traditions culturelles techniques. Et puis, il y a des autres travaux. Il y a un travail qui va sortir bientôt sur la génétique et sur la nouvelle série des dates, et sur lesquelles il y a la possibilité de confirmer que le second mésolithique et le premier néolithique appartiennent à la même famille génétique. Mais pour l'instant, au niveau du lithique, on peut parler d'une tradition culturelle, mais pas évidemment d'un contact. C'est pour ça qu'on ne parle plus de transition.
Okay, so we have another question. Oliver Craig is for you. So the question is, how is possible to detect beer residues on pottery, and how an underwater context like La Marmota can affect the presentation? Yeah.
Yeah, who asked the question?
Okay, the question is, how is possible to detect beer residues on pottery?
Right.
And how an underwater context like La Marmota can affect the presentation?
Well, so I think it's very difficult to identify beer residues on pottery. There have been some examples where people suggested it, but direct chemical evidence, I think, is really difficult to distinguish cereals in pots cooking from actually fermentation. Maybe the occulobotanical method would be better, looking for evidence of sprouted grains, and we see this in later periods, but in the Neolithic, as far as I know, very little evidence of that. So I would say very difficult, even with good preservation conditions like La Marmota. It'd be quite difficult to actually be sure that you're looking at fermented barley or wheat within the vessels. But wine, I think, has got a lot more potential.
Yeah, because now the question is, how can we distinguish the fermentation and the way?
Can't is the answer. We have no clear markers for fermentation, so we can never be sure. However, if we find vessels which have high concentrations of this molecule, tartaric acid, in relation to this other molecule, and we don't find evidence of other types of fats or oils, then I think, you know, we're looking at grape juice. Do we stop there, or do we say probably this is fermentation? This is wine, and I think we probably do have to do that. We have to say, you know, this is fermentation. Other things we can do is perhaps look at the DNA of some of the grains of all the wine, and then maybe have a look at some of the cultivars and see whether they've been under some kind of selection or domestication, which could be used to imply that they're being grown specifically for wine production. But really, I think the residues has little to offer. It has something to offer, but to tie it to the vessels. But I think the major part is still the archaeobotany. You know, so like Amy's work is very important there.
Yeah. So maybe Amy, talking about fermentation or the identification of fermentation in the archaeological remains. Can you hear me?
I'm so sorry. Could you just repeat?
Yeah, because we are speaking about this fermentation process,
Yes.
identification in the organic recipe analysis, and of course has been suggested with the archaeological remains. So is the evidence in the archaeological remains quite obvious, or is always difficult to say the kind of fermentation?
There's actually been a sort of surge of work on that, on what can be done both to detect germination in grains, including with SEM. Can you still hear me?
Yes.
And the key project that I'm aware that's produced really important work on this is Plant Cult. So the ERC project that Tanya Valamotti has been running at the Aristotle University in Thessaloniki, and the website is great. So there's quite a comprehensive treatment of the relevant publications.
Yeah.
Okay.
Can I ask Amy?
Yeah, of course.
But I mean, do you find any evidence for those kind of typical? She's got crushed bitter seeds within quite a few of her assemblages in northern Greece, and I was wondering if that extends also to the Peloponnese, or if that's been investigated.
I mean, the sporadic opportunities I've had to look in the Peloponnese, I haven't found it. But as it happens, this past summer, partly because of the pandemic and not doing normal field work, we've been looking at some material that was passed on to us by Robin Denel from Chevdar, one of the classic kind of early Neolithic Bulgarian Tell archaeobotanical investigations. And it is full of crushed grain. And it's also got germinated, I think it's naked barley grain. So I think we need to go back to some of these early assemblages and look again. I think that it's probably much more widespread than we realize.
Yeah, thank you, Amy. So, any questions?
I can repeat the question if you want.
Yeah, it sounds good.
Yes. Just ask me and I'll repeat.
Okay.
So why is there so much interest in wine and beer? Well, I mean, I guess the answer is, I mean, Amy's been working on, how all these plants have been cultivated, but what the side that we're interested in, I guess, with the pottery is actually how the consumption practices have been played out. So what these grains have been used for, how they've been mixed in pots, and I think fermentation has that role to play there. When we start to see, why would you put cereals in a pot in the first place? Why do we start to see these plant products going into pottery? And people have suggested that the reason for that is for producing these kind of special foods or for the process of fermentation. If you think about making bread from wheat, you wouldn't necessarily need pottery, you should be really involved in it. So, when you start to see large amounts of cereals present in some of these vessels, you can think of porridges and gruels and things like that, but I think also fermentation is something that we need to consider. Yeah. I've got a question for Amy.
Okay. Amy, we have a question for you, sorry.
Which is the variability in the nitrogen isotopes in the plant remains that you see. They're very variable, aren't they? I mean, it's that even then, you're seeing lots of variation even within one assemblage. And I was wondering, is that to do with, I don't know, the variable in the growing conditions year on year? Is it to do something with preservation that's altering the nitrogen isotopes? Because it makes me think, actually, the animals are much less variable. And maybe they're averaging because they're eating lots of different things, and that's the reason. The human's less variable as well because they're averaging. But just looking at a few seeds and trying to base our assumptions, and that must be very difficult when we have such a large amount of variation. And I wonder if you've done experiments to find out what causes that kind of variability.
No, that's a great question. There is a lot of variation, and it's even greater if you do single grain analysis. So the results that I was showing you are from bulk samples of normally 10 grains each, partly because we're trying to average that out and to compare it to modern growing conditions that we similarly sampled. But the variability we're seeing it's consistent with the variability you get in a modern, even experimental growing condition, meaning someone is standing there with a lab coat on and a clipboard doing the same thing, you know, month on month, year on year, and you still get variability. So I think the danger here would be to look at just a few, say, single grains to try and make inferences about growing conditions. If you're going to do it on a big assemblage where you can throw a lot at it. And then you can understand, as we've begun to, and you've picked up on that immediately, how variability is then averaged out in the consumers. And sorry, you mentioned preservation as well. We're trying to exclude preservational issues in the way that we select the grain. So we now do this thing where we even do a cross section of each grain and look at it, because we've been looking at experimentally charred grains under SEM to get exactly an idea of the conditions under which they're carbonized, because charring definitely has an effect. So you've got to be careful. It isn't in that sense like collagen, where if it falls within a certain preservation window, you're fine. You really have to have kind of archibotanical, nerdy eyes on it.
Yeah, thank you. Any questions, comments?
So we got there. I have a question for you at the point.
Yeah.
You were mentioning the analysis that would be mainly to
sorry?
On your presentation, we were just thinking about the lithic analysis and the main system, but you also showed the grinding stone and the down and start analysis and grinding stone, because it also can give you a lot of information about production of in which materials are used. So if you did
okay, I'll ask the question.
Yeah.
And you can answer.
Okay.
So you're asking about whether the grinding stone material, and you know there's work's been done on the grinding stones, and you're saying what the utility of that is for looking at starch on the grinding stones to understand their use and other kinds of markers. I don't know if you want to answer, Miriam. We've done very little work on grinding stones. Work has been done on starch. I don't know, the starch community is kind of divided about how well starch can be trusted from that source. You know, starch is everywhere. And I think by taking a proper sample, maybe from the grinding stone surface, stuff that's embedded within the porosity of the grinding stone and the exterior surface of the grinding stone, maybe we can make some inferences. But I would also be quite kind of wary of using starch in that way. Of the specificity we can actually get down to with starch identification is also a little bit problematic sometimes. We get changes in the starches. I'm not a starch expert, but you know, we're kind of a bit skeptical about it, I think, in the archaeobotanical community. Amy might want to comment, I don't know, about starch. So thoughts?
Well, I just would underline everything you've said, and that, you know, all of the control sampling is so important for comparing these artifacts with surrounding sediments, and some quite convincing work has, I think, been done, but always framed then in comparison with, as you say, the ambient starch that is present anyway.
And so there is a project going on now, a new project, to try and look at a molecular identification of grinding stones, material within grinding stones. So I think Tobias Richter, who's in Copenhagen, he'll be working on it. And we've also got some grinding stones from Neolithic sites in Scotland, so two very different environments. And the DNA guys and the proteomics guys are going to try and do some tests to see whether they can do some identifications in that way. But I would guess, as Amy says, the same problems would occur, and you really need to be able to take samples very carefully from the actual artifacts themselves.
more global question, observation. We used to work on the transition of Mesolithic, Neolithic, and Septiembre. That's truly it's about North Mediterranean. And there's a lot of, and then there's a lot of leaks about North Africa. It could be the blended, trapeze blended. It should be the rock art. And according to you, there's next year, there's the hope to have to also question the hypothesis of a transmission of Neolithic by North Africa or no.
Shall I ask the question? You could hold me. The question was, a lot of work research is focused on the northern Mediterranean, looking at the dispersal of the Neolithic and the artifacts. But what is the potential for looking at the southern Mediterranean African coast and building that into our interpretive framework? So,
Yeah, I'm completely sure that the future of the research is in the north of Africa because we just have the picture of the north. So it's quite important to have all the basins quite well recorded. So we were speaking about that last week with Giulio Lucarini because several projects are now developing in the north. And even Manolo made a nice archaeological survey, in the island between the Iberian Peninsula and the north of Africa in the Islas Chaparinas, with a very nice cardial site that, of course, is showing something is happening, in the Gibraltar Strait. So I think to have all the picture, all the scenario in the Mediterranean, we need to improve and develop these projects in the north, because it's not, of course, it's just our European vision. But Giulio Lucarini and other teams in SATE are developing this kind of projects in the north, especially in the Mediterranean area on north of Africa. There are not several projects in the Atlantic coast north of Africa, but it's a quite important place also, because we have the Canary Islands and everything to compare. So, but it's true that in these following years will be a quite interesting focus of the study.
Donc, à notre avis, il y a une dynamique au 7e millénaire qui peut avoir des liaisons avec le nord de l'Afrique. Et puis, il y a une dynamique néolithique qui est liée au bloc oriental. Donc, c'est en étudiant les sédilithiques et en croisant un peu les données avec les systèmes économiques, la partie de la génétique, tout ça, c'est qu'on peut se poser une question. J'ai réfléchi sur la grotte de Luzzo, par exemple. Si les sites sous la côte de la Méditerranée pouvons avoir développé des modèles différents, et donc, si on peut parler d'un contact entre le dernier mésolithique, les premiers néolithiques. Mais à ce moment, je crois que c'est un sujet encore à développer, comme, à mon avis, on peut encore se poser la question sur la date très ancienne de l'horizon à trapèze et à l'an de la grotte de Luzzo, qui est un peu en contraposition avec les données chronologiques qui viennent de l'est de la Méditerranée pour le 7e millénaire. Mais à notre avis, il y a donc deux dynamiques différentes. 7e millénaire, l'âme à trapèze, et puis 6e millénaire, c'est toute la dynamique impresso-cardiale, un peu comme Didier Lunaire l'avait construit.
More questions, comments, anything? I have a question for Eva. Eva, thank you so much for the summary about ancient DNA, because you know it's quite difficult just to follow the literature, and the bibliography is like a mess. So in one moment you are completely lost. So thank you so much for the summary. It's quite interesting, but because all of us, we usually study the early Neolithic, so the transition between the Mesolithic and the early Neolithic, looking for this contact between hunter-gatherers and farmers. And the DNA clearly shows that the contact, at least the genetic contact, was in the middle Neolithic, because you can see this mix between hunter-gatherers and first farmers. And I agree with you, because it's quite interesting just to check the social complexity, you know, a more intra-specific study in the archaeological sites. And these kind of archaeological sites are quite frequent in the late Neolithic. But how can we explore that in the early and middle Neolithic? Because these kind of contexts are quite difficult. So how can we explore that?
Oh, that's a tough question. I'm not sure if I have an answer for that. Yeah, it's right that it seems that in certain places, at least, this relationship between the hunter-gatherers and, well, hunter-gatherers pre-Neolithic populations and Neolithic populations, it took some time to unfold. Yeah. These contacts, they were probably initiated during the Middle Neolithic, and then you see this steep increase in pre-Neolithic ancestry during the late Neolithic. So it's just still, I mean, there's not like a satisfactory explanation of how these types of ancestry survived over the time, and how they just, it took so long for them to like be present in an admixed form. There is an interesting like paper from Barbara Bramanti, Ruth Bolongino, sorry, that she talks about like parallel societies, and she establishes a parallel between the economy, the subsistence economy of that it was maintained separately. She uses stable isotopes and ancient DNA. So I believe that the answer is like to your question about Middle Neolithic contexts. It's a multidisciplinary approach. I don't think that ancient DNA, even with like high resolution serial sampling, meaning that you try to follow the same population over time, that's not possible in many contexts. Yeah. But just trying to find the answers in like a combined approach of archaeological science. I think, for example, stable isotopes of mobility, they are quite useful in this regard, being able to like separate like first generation migrants from the genetic signature. The signature that you see in the DNA is like much more deep in time. So we cannot really pinpoint if the individual is a first generation migrant or that admixture happened like a few generations ago. So we can just do some calculations, but that's it really. So just the combined effort, I think it's just what gives the like a more like satisfactory answer, but not just from ancient DNA, stable isotopes and other disciplines in archaeological science, but just archaeology itself. What do we know about this context? Do we have reliable dates? What's the history of the site? So I think it's just a multidisciplinary effort, and it would be just a question of like shifting the view, and that, as I said, that's happening already, at least within the ancient DNA field, from like big geographical areas to like discrete sites, because we see in some instances that these contacts, they happened like in an uneven way in different places. Like for example, Abri Pendimont, it seems that the individuals that they lived there, they had like a higher pre-Neolithic ancestry than other individuals in the same like cultural complex. So there seems to be, you have like the specificity of the site, and that needs to be studied properly alongside other sites of evidence that they are not ancient DNA. Yeah, I just think that's the way to go.
Yeah, Yeah, I agree with that. Thank you. Any questions, comments, anything? No? So if we don't have more questions, yes, I'm sorry, we have another one.
So it's about the later movement of population, not in the beginning. So most of the ancient studies are focused on from Mesolithic to Neolithic and early Neolithic. But how much done of ancient studies in late Neolithic and calculus? Because evidence of new impacts from different parts of the world, I mean, from Anatolia again to Southeast Europe and from other directions as well, especially from it. So my question is whether there are new studies about late Neolithic calculus movements of population and whether that information, where these people are coming from.
Yeah. Eva, we have another question. Did you?
No, I just picked like the sound is not really.
Yeah, I know. I'm sorry.
Good, so if you can just summarize.
Yes, I know. We have another question. So most of the ancient DNA studies have been focused on the transition Mesolithic, early Neolithic, but we don't know a lot about the middle, late Neolithic. So the question is how much information is available about the population movements during the middle and late Neolithic?
There is considerable amount of data on middle and late Neolithic, and in some cases where there is no early Neolithic samples available, or individuals that they have produced DNA because of preservation. Some of the, especially the middle Neolithic samples, have been used as proxies to try to establish what happened at the beginning of the Neolithic. But overall, obviously there's like there is an increase in population in most places, so which means that there are more samples to analyze. But the trends, I mean, overall, just speaking at continental level, we tend to see like some sort of like genetic continuity from during the early Neolithic up to the middle Neolithic. And then in the course between the middle Neolithic to the late Neolithic is when things start to change. And then with this increase in pre-Neolithic ancestry. So, but again, that's what we see with the data that we have now. And I agree that maybe if we just like explore and analyze more samples, other interesting patterns will emerge. Had to do with the like later consolidation stages of the Neolithic during the middle and the late Neolithic. There's also like an important like wealth of information about what happened at the end of the Neolithic and at the beginning of the Bronze Age, with other like population movements or like ancestry arriving from the east, but that's another complex and different story. But yeah, there's like several papers focusing on like more middle Neolithic and late Neolithic populations, and also just seeing how these early Neolithic populations, what happened with them. So there's some papers focusing on the Near East movements during the Chalcolithic. There are some others on Iberia. There are some others on Central Europe. So the evidence is still a bit patchy. But and yeah, and then just recently there is also an interest for studying like social structures, and the contribution of ancient DNA has been just seeing to see how individuals, they relate biologically to each other. So if you can infer patterns, any like patterns of like let's say, well, marriage, I know it's not the right term, but just how they mated with each other. If we can talk about matrilocality, pratrilocality, so that's another dimension of the ancient DNA work, and that there is like a Middle Neolithic and Late Neolithic populations, they form like an important body of data of what's being studied at the moment in terms of like more social structures.
Mm-hmm. Yeah, thank you. Some more questions? Something? No? So if we don't have more questions, thank you everyone for your comments and for replying all the questions we had. So we have now lunch time, fortunately. So we will come back at 4. Yes, at 4 this afternoon. So thank you so much for joining us, and we have the lunch time. Enjoy your sandwich in your countries. Thank you.
Matteo, siamo in diretto? Matteo? Bueno, hacemos una cosa.